So as I already mentioned, we will have with the keynote presentation from Professor Yan Hao. He's going to give a presentation titled Animals in Biomedical Research, Success, Challenges, and the Way Forward. Professor Yan Hao is a specialist in comparative medicine and laboratory animal science and welfare. He has been active in animal research for more than 50 years and supervised dozens of postgraduate students from different corners of the world, including 5 scientists from Kipre, which was formerly IPR. For more than 35 years, he has been a professor at the universities of London, Uppsala, and Copenhagen, where he was responsible for the centralized animal care and use programs. He has published about 500 scientific publications. Dr. Yan Hao is now semi-retired and just doing what he likes the most. Let's give him a round of applause as he comes.
Dear organizers, thank you very much for inviting me to give a presentation on the importance of animal research here at this conference. And ladies and gentlemen, it's an honor and it's a privilege for me to be given this opportunity to talk a little bit about the importance of animals in biomedical research. So that's the title of my talk, and as you can see, we could spend many days discussing this, but I will just give you sort of a brief presentation of some of my views when it comes to animal research. It's interesting that we use animals to study human biology. Many of us are not really interested in animal biology. We are interested in human biology, but for many reasons, it is often impossible to study humans when it comes to, or when it involves procedures that are associated with pain. And people have appreciated the importance of animals in research for many, many years. This is a picture of a famous Danish physiologist who said to his students when he lectured to them that, you know, man is not really man. Man is composed of different species. So we have studied muscles in frogs, and we have studied the brain in cats, and nervous actions in grasshoppers, and what have you. These studies have not been performed in man. They have all been performed in animals, where we have used animals as models instead of using ourselves. And he was one of the founding fathers of the scientific discipline, which is called comparative medicine. And comparative medicine is very briefly defined as the study of both normal biology, normal and abnormal structure, and function, and really very much about choosing the right animal model, because as August Krogh said, there will always be an animal that is suitable for your studies of human biology. I was also told to talk to you a bit about successes. I mean, it really is a success history, because we, as you saw before, almost all what we know about our own biology, we can thank the animals for. And successes, look at these numbers of Nobel Prizes given to scientists who have used animals, been dependent on the use of animals in their research. So there have been 232 recipients of the Nobel Prize in physiology and medicine, and 195 of those were reliant on the use of animals. So it is, I mean, the importance can't be overstated or overemphasized. Let's see. That was this one, we've seen that one. And look at this one. This is the king of the castle right now when it comes to laboratory animals, the mouse. And we owe the mouse a lot of gratitude for its services in science. And you can see here again, I know you can't see the details, you can't read it, but it's a long list of Nobel Prizes that were completely dependent on the use of the mouse as a model for ourselves. Excuse me. So if we look at the definition of an animal model, it's on the screen here. It says that it's a living organism in which we can study either normal biology or behavior, or in which we can study an either spontaneous disease developing in the animals or an induced disease where we deliberately make them sick to serve as a model for man. Now, it's very important here, of course, that we choose the right model. And that is not always so easy. As we talked about a little while ago, we have really studied many different animals. I know that there are certain animals which are far more popular than others. Take the mouse, the rat, the dog, the pig, and the non-human primate. But we have really used a lot of different models throughout history in different species. Here I have chosen, and for this brief presentation, I have chosen to focus on the non-human primate. So the examples I'll give you are really non-human primate studies. I'll go back 50 years when I started my research career as a medical student, where we were investigating human pregnancy proteins and the functions and what have you. And we used mouse models. Mouse models were actually fine, but there were some problems with using mouse models because much of that research was reliant on immunochemical assays, immunoelectrophrases, ELISAs, and you all know about these assays. And the problem here is that antibodies raised against human proteins, they normally don't work with animals or with the same proteins in mice and rats. So you can't really use all the commercial antibodies available against human proteins. What you can do is involve the non-human primate, because the non-human primate is biologically so close to us that most antibodies raised against human proteins, they will also work in the monkey. So you can quantify the protein in the monkey, and you can be fairly certain that it's the analogous model that you're working with. So here, the non-human primate was very important for this part of the research. But let's go to tropical infections. This is also an area where non-human primates have been essential in biomedical research. And very often when it comes to diseases, infectious diseases, it is necessary to move up in the phylogenetic tree simply because lower ranking species do not get sick, they do not get infected. Here is an example where we worked with chronic schistosomiasis in baboons, and this was Dr. Niakundi from Kipra who did this research, very important and interesting research, looking at how tropical infections, how they affect each other. Because normally when you have a person suffering from a tropical infection, she will very often suffer not from one, but from 2, 3, 4. And of course these infections, they have an impact on the course of the other infections. So it's actually very complicated research, of course, but very important research. And Dr. Niakundi did some very good work. Also on schistosomiasis, Dr. Farah, also at that time at Kipra. He also used the baboon. He also used mice. But mice are actually a fairly good model for schistosomiasis in many ways, but it's so small. So just one worm in the liver developing, and you really can't use it anymore. Whereas the baboon, the disease develops very, very similar to the way it does in us. So that's very important. He also focused on a very important aspect, and that is to refine the techniques and have a focus on the animal welfare. Because previously, many protocols for when you do this, OECD protocols and the European Pharmacopoeia, they had death of the animal as an endpoint, which is obviously not good for the animals, because before death, there's suffering. So he did some very important work to develop new guidelines, new, more humane guidelines, where death was not the endpoint for the animals. Another very good scientist from Kipra, he used vervet monkeys because he was studying human trypanosomiasis and the influence on therapy, on the cause, the etiology of the disease, and Dr. Ngotho did some very, very good work here with models. I'll tell you a recent example, and this is this is interesting because this is again where the animal develops spontaneously a disease where we say, you know, hey, we can use this as a model. This was Dr. Darrisman, who was also a PhD student of mine, and we did a project together, and it all started by his technicians in the animal unit. They told him that some of the old monkeys, they couldn't remember where they had hidden their treats. You know, they get raisins and things when you pass, and if they can't eat them straight away, they hide them. Some of these monkeys, not all of them, but some of the old monkeys, they went looking for them because they couldn't remember where they had put the treats. So we wondered, you know, could this be a spontaneous model for human senile dementia? And lo and behold, we looked at the important hallmarks here: short-term memory loss, lesions in the brain, as you are familiar with, biomarker changes in the circulation and in the cerebrospinal fluid. And we could tick all the boxes. So we have here what seems to be an ideal spontaneous model for a very, very important human disease that we all fear. Some of us more than others, of course. Those at my age, we fear it a lot. And all you young people, you may not think about it, but it is a terrible disease. I think, yeah, talking about all of this about animal models, I wondered, you know, who were first to use an animal model? Can you guess who I think it was? Mr. Noah, when the world flooded and everything was covered in water. And he wondered, when can we ever, you know, come back to dry land? And instead of sending one of his sons out in a little dinghy or rowboat, he said, you know, let's use an animal model. Let's use a pigeon. And that's what he did. He sent out pigeons, and eventually they came back with an olive leaf. And he knew then that the end was near of this misery, and they would soon land on dry land again. Well, I'm also asked to talk a little bit about challenges. And I think there are many, many challenges facing us using animals as models. One is the translatability of the findings, whether they are relevant, our findings are relevant for the human. In the old days, we talked about extrapolation of the results, but it's really the same, it's just a newer term. And I think there's very often a problem here, and I think that people tend to misuse the term animal model and sort of overreach in the many aspects of a model. Because sometimes you're really just working with one mechanism and people tend to call it a model. And then sometimes you work with a very complicated human diseases. that are defined, by if the patient has protein in the urine, if the patient has high blood pressure, and if that, then we call it preeclampsia. But an animal model for that is difficult. So I think people should be careful when they use the term animal model. There's also been a lot of criticism recently about the lack of reproducibility, particularly perhaps in behavioral studies. Behavioral studies are very, very difficult, and pharmacologists, they use them very often. But unfortunately, the predictive value of behavioral models is fairly low compared to models of the hematopoietic system or gastrointestinal. Once we're talking about the brain, it becomes really, really difficult. And the question is really, is there an animal model of human depression? I know there are many models described, but do we know enough about the human disease to be able to determine whether we have the possibility to develop a relevant model in an animal? Then we have something fairly recent, and I'm sure you have been aware of that, particularly if you follow LinkedIn and so on, and that is that there are many fraudulent articles. This is really, this is really bad. I think that when you do these big meta-analyses where people have manipulated illustrations and histological slides, then you find out that perhaps at least as many as 5% of all publications are fabricated, are fraudulent. Excuse me. And then, of course, we have ethical concerns all the time regarding animal welfare and suffering. And this is an ongoing process. We try consistently to improve the conditions for the animals. To the public, I think the public very often have a skewed picture of what animal research is, and they think, when they think of animal research, they think of testing. They think of safety testing, and they don't consider that the majority of the research is actually basic research and translational and applied research. Excuse me. These are figures from European statistics, and you'll see that regulatory studies and routine production, they account for less than 20% of all the animals used, whereas about 70% are real science, or are basic science and translational and applied research. And that the public very often has this understanding that animals are not really necessary and they can be phased out within the foreseeable future, I think is a big worry to many of us. And then, of course, we have people who try to disturb scientists who, as a special interest, wants to stop and ban animal research. Sometimes they're very violent, and they put bombs in veterinarians' cars, and they do all kinds of nasty things. The interesting thing here is that they do have an impact, of course. They are very good at lobbying, particularly sort of the more civilized of them, because there are many respectable animal welfare organizations, and they are very effective in lobbying, and they use many resources to lobby politicians. So there's no question about that they have an impact on legislation also. Whether we should be critical of that, I'm not really sure, because at the end of the day, I think that the changes you see to legislation improving conditions for animals, I think we owe at least some of that to the people who are fighting for animal rights and whatever they call themselves. It's interesting, we talk very often about ethics. You know, ethics, it's an ethical problem when we use animals. And yes, it is an ethical problem. It is, because we cause them sometimes pain and suffering. If we study pain research or study pain in humans, it's difficult not to have pain in the animals. But there's another very interesting ethical dilemma, and that is if we suddenly should stop something happened to the pictures here. Could someone fix the pictures, please? So it works again. Yeah, but if you could fix that, then I'll continue talking. There's another problem, ethical problem, which is in fact, I think, not really discussed much, but which is very important. And that is, what would happen if we suddenly banned the use of animals in research? As Charles Darwin already stated, he actually said that whoever would hinder the use of animals in biomedical research, he called it physiology, they were committing a crime against mankind, simply because they would be taking responsibility for depriving our children and future generations of treatment and prevention of important diseases. That is very rarely discussed, but it is, of course, very, very interesting to bear in mind. If you should ban the use of animals in research, you take on a big responsibility. And it's not very unlikely. I mean, they vote about this in Switzerland from time to time, whether to ban it or not. But the industry is so strong that they don't ban it, but it does happen in certain parts of the world. I think the state of Oregon also voted about this, and I won't talk about history. There are countries in history who have done this, but that was a shady part of history. Now, you could ask, many people say, we are so clever nowadays. I mean, we have AI, we have quantum computing coming up in a few years, we have in silico, we can build organs artificial. Why is it really necessary to use animals in biomedical research? The best answer to this question, I think, I found in a little publication on behavior by Martin and Bateson, two Cambridge professors, and they said it really has to do with levels of biological organization. We as scientists, we study biology at all levels of biological organization: molecules, cell organelles, cells, organs, and then comes the individual, and then comes groups where the epidemiologists study things. But there's a lot of information that you can't get about the intact individual. You simply can't get that from looking at other levels of biological organization. It's like not being able to study the real thing. So I think that's why we're always going to need animals. So what do? We have many challenges, many issues of different kinds. I think for those of us working with animals, I think it's important that we appreciate that there is a tremendous positive development in both reduction and replacement. I mean, we have non-animal methods, or whatever they call it, abbreviated NAMs. We have cell cultures, we have organs on chips, we have artificial intelligence, and we have all these things available. In my mind, there's no question this will reduce the number of animals needed in research. Now, for the animals that are needed in research, I think the most important of the 3 R's is refinement. That we continuously strive to improve the conditions, the husbandry, the care, and use of animals in research, and establish true cultures of care and openness in our use of animals. Let's take, with respect to refinement, let's take a look at what has happened quite recently. We have moved from fairly small cages to larger cages. That's a general trend across all species, that the cages are now much, much larger than they were 30 years ago. And we have focused a lot on social housing, both in the American Guide and in the European legislation. It is a requirement that animals are, as a rule, socially housed when they are social species. So we move from single housing to group housing. To have animals in a good environment is very, very important, and it is extremely stressful to be single housed, particularly if you come in from the wild. And a very good scientist actually worked on this issue. Again, a Kipra scientist, so you should be very proud. He found that when you put African greens into little cages coming from Kibwezi, living in the wild, then some of the animals, they're very, very susceptible to the stress. And you see that there's immune suppression in these animals. They have lesions, not just in the adrenals and stomach, as you might find, logical, but also in the hippocampus, with big clearings in the hippocampus, and persistently elevated levels of cortisol in the circulation. And Dr. Kagira, also from Kipra, he showed that from you put an African green into a cage, it takes 6 months for all the biological parameters to come to a new steady state, which is really remarkable. So, good housing conditions are very, very important when you have animals in research. So group housing is the way to go. Training the animals so they become confident and they like to interact with people is important. And of course, to desensitize them to procedures, make mock procedures and train them so when they are subjected to the real thing, it's no surprise and it's not painful. Because pain is closely associated with fear. So pain is a perception of the mind. So if you're afraid of something, the procedure will be painful. Take dentistry as an example. If you're afraid of the pain when you go to the dentist, and really afraid of the dentist, there's a very good chance that it will be painful like anything. If you consider it a laugh, then it will not be painful at all. This is an unusual circumstance for this monkey. It was in a colony when we were in Uppsala, and they preferred always to be outdoors, no matter what the weather was. Nothing beat the outdoors, which is another strong argument for having the animals in their natural habitat, natural environmental conditions. Metabolism cages and all cages that restrain the animals and transport is very stressful for non-human primates. We have shown that in different projects led by Dr. Paramastri from the IAB, and she demonstrated that metabolism cages are initially stressful, but if they are confident and well-adapted animals, then it's a very brief duration. So here you can see they were put in metabolic cages, and it took only really a day, and they had adapted to these new conditions, and that was because they were well-adapted animals. When you transport them, some people transport them in individual crates, some in double crates, and some where you have a separation between the two animals with a little window. And that is very, very important, because being transported alone at convoluted routes by truck and by chartered aircraft and what have you is very stressful for monkeys. And here she did a study looking at the behavior of the animals depending on whether they had been transported alone or with a companion. And it was clear that it is far less stressful to travel with a companion than to travel alone. Aggression after transportation is another good sign of stress, and here you'll see that that when you have been transported single-housed, then you maintain an aggressive approach to people for a very long time, whereas if you have been transported in a pair, that's the red line, then you'll see that stress is of shorter duration. Whoops, did I go one too far? Yeah, and then I would like to talk a little about Professor Shapiro's research, also a member of the IAB. He studied whether training, positive reinforcement training, would actually lower stress and stress-associated parameters in chimpanzees. So we looked at 133 chimpanzees during their annual physical, where you did chemistry and hematology and blood pressure. And very interestingly, we found that the animals that were trained to voluntarily present an arm to be anesthetized for the annual physical, they were far less stressed than the animals that had to be chased and involuntarily were anesthetized. So you can see a number of stress-sensitive parameters here, clearly indicated that positive reinforcement training reduces stress. So very important. Now I'm almost finished. I don't know how long time I've spent, but some concluding remarks. I think it's important that we have more transparency. We must get the public on our side and show them that we really care about the animals, that they are treated as well as we can. And we talk a lot about these years that we must have a culture of care. And that must go for all staff in a program, that we're all interested in improving the conditions for the animals. And also train the animals, desensitize them to what is coming. And I think, unfortunately, NHP research is actually particularly threatened. And in terms of legislation, the European legislation actually prohibits the use of non-human primates for research unless we use them to study an important debilitating disease in the human. Then it's okay. But you can't do all kinds of non-critical studies in primates. And that leads me actually to advocate for, and I've done that for many years, I think it's important, non-human primate research is being concentrated more and more in big centers in Europe, in the US, in Asia. And I think it's very important that this happens in resource countries, in countries like Kenya. It really belongs here, because then you don't have to transport the animals, which is always stressful. They don't have to experience a change in climate. They are on their home turf. So I think that is really something we should argue strongly for whenever we can. This is my, I think this is my last slide. And I think a very important message here is that it's important to be kind. It's important to show kindness to the animals, just as you would do to your pet. These animals are no different. They also deserve to be kindly treated. And, you know, if you're kind to animals, it actually sort of shows that you are also kind to people. And if you're kind to people, you're kind also to your fellow countrymen, other nations, the whole world. It all starts with kindness. So, thank you very much for listening.
Yeah, thank you very much. Let's appreciate Professor Yan Hao once again. I heard him say that out of the 229 Nobel Prizes that have been ever awarded, 195 as a result of working with animals. So I'm hopeful that I will get one very soon. And all of us who are here, because we have interest in this kind of research, I think we are moving very well. So we skipped a session in the morning. We want to return to the session. The distinguished guests have arrived. Again, my name is Dr. Atunga Nyachio, the Director of Research at Kipre. I will invite Madam Pauline from the hotel for a 5-minute presentation on security, safety of the hotel. Then from there, we will go back to the program that we skipped in the morning. Pauline.
Thank you, Doctor. Good morning. Good morning, ladies and gentlemen. All protocols observed. I'm Paulina Cheng from Safety and Security, and on behalf of the hotel management, a very warm welcome to Emara Ole Sereni, Nairobi. Where we are, the meeting room is called Loita Siana. In an event of an emergency and evacuation order has been given through the speakers, we all shall use the fire escape doors behind, marked clearly, to fire assembly point that is located outside the hotel main parking. Today, there's no planned fire drill, so we expect 0 interruptions during our session. For medical attention, the hotel has a clinic that operates from 8 to 5, and from 5 onwards, we have doctor on call. We have a smoke designated zone that is outside the glass door or behind this terrace facing the Nairobi National Park. That's where we have smoke designated zone. Washrooms are outside the 3 elevators. You walk straight, then you turn. That's where we have gents, ladies, and people of determination. Your lunch will be served at level 3. You'll use the 3 elevators, 3rd floor, but before you go there, you'll be issued with lunch vouchers. That's where we'll have your lunch. During that time, security will be here to make sure your items are safe and secure. For purposes of access control and identification, please make sure you wear your badges at all time. And it was also important to make a point of knowing the neighbor that is seated next to you. Once again, thank you so much for choosing Emara Ole Sereni to have a productive session. Over to you, Dr.
Thank you, Pauline. Let's appreciate her once again. Now, I want to invite the Director General, Kenya Institute of Primate Research, Dr. Peter Gichui Muidera. Let's appreciate him as he comes.
We had to rearrange the program because we didn't want to start the formal session without the chief guest, who is now with us. So the representative of the Cabinet Secretary for Health, Principal State Department for Public Health, PS, Madam Mary Muthoni Muriuki, the members of the Departmental Committee for Health, led by the chair, Dr. Nyikal, the Board of Directors for Kiple, led by Professor Josephat Muriba, members of the International Advisory Board for Kiple, led by the chair, Professor Yan Hao, partners, collaborators, delegates, organizers of the 20th Kiple International Scientific Conference, ladies and gentlemen. It is with great pleasure that I welcome you to our annual health conference, hosted jointly by Kenya Institute of Primate Research, Mount Kenya University, and the Society for Biomedical Biotech Research. Your presence here today is a testament to your commitment to advancing health and well-being in our communities. This conference serves as a vital forum where we can gather to share knowledge, discuss emerging trend, and explore innovative solutions to the pressing health challenges we face. The world of health is ever evolving, and it is our responsibility to stay informed and connected. And the theme for the conference today, tomorrow, and Friday will be transforming health through innovative research. This theme is very relevant to this country because for the first time over a very long period, Kenya has failed this year to feature among the top 100 innovators, and this is not very good for us. This calls for seriousness in creating and supporting research to improve the innovation ecosystem. Collaboration is the cornerstone of progress. This conference brings together a diverse group of professionals, including researchers, practitioners, policymakers, and advocates. It provides an international forum for sharing latest advances in science and technology, and support research training and human capacity development in the biomedical and biotechnology fields in Africa and beyond. I would like to take a moment to acknowledge the hard work and dedication of all those involved in organizing this wonderful event. Your efforts have made it possible for us to come together in this important dialogue. Additionally, I would like to recognize the invaluable contributions of our speakers, whose expertise will enrich our discussions. Before I proceed, I would like to take a moment to honor the memory of one of Kiple's founding scientists, and this is the late Dr. Jim Ells. Dr. Ells was not only a brilliant researcher, but also a visionary scientist whose immense contribution and unwavering dedication laid the foundation upon which Kiple now stands. Under his leadership, this Primate Research Center was born, and he served as the institution's director from 1975 to 1989. He passed on in May 17th this year in Brunswick in the US. But he left behind a legacy of excellence, compassion, and scientific integrity that continues to inspire our work. In recognition of his enduring legacy and impact he made on this institution, I now want to kindly invite all of you to observe a moment of silence in his honor, and we can kindly start. Thank you very much. You may be seated. May his memory continue to live on through the work we do every day at Kiple. As we move on through the sessions, let us keep in mind our shared vision to create an equitable and accessible healthcare landscape. The ideas exchanged here have the potential to inspire action and drive meaningful change in the health sector. I appeal to the African governments, starting with Kenya, to invest in science, technology, innovations, entrepreneurship, and manufacturing, because these are the drivers of economies and they stimulate growth, hence lowering poverty. And now we'll be doing a presentation tomorrow where I'll give examples of where this has happened across the globe. In closing, I wish all of you a very fruitful and inspiring conference. May the knowledge and connections you gain here empower you to make a positive impact in your respective fields. Thank you for being here, and let us embark on this journey together towards a healthier future. Thank you very much. It's now my honor to invite the chair of the Board of Directors for Kiple, Professor Josephat Muriba. Karibu, Chair.
Morning, all of you. to the chief guest, the PS, PS of Health, Public Health and Professional Studies, KIPRE Board of Directors, the Committee of Health of Parliament, led by Dr. Nyikal, AI, BBC, Mount Kenya University, members of KIPRE International Eminent Science Advisory Board, led by Professor Jan Howe, KIPRE and other local and international scientists. I am honored and privileged to welcome you to this biotechnical conference of transforming health and informative research. Two things are achieved at this conference. First, there is the flow of knowledge from the north to the south. We always say the north is advanced and the south is advancing. And somewhere we meet and there is transfer of technology. And I think this is very, very important. The other thing is that we have now tended to have virtual meetings. And virtual meetings actually don't transfer as much knowledge and useful interaction as when we have got actual physical meeting like we have today. I know that for the last 2 days, the IAB has been at KIPRE, and they have actually gone around, seen quite a bit of what is happening there, and advised. And I'm quite sure that actually your physical presence makes much more difference than when you actually advise from far. Recently, a president of one country said, if you people agree on a ceasefire, I will come personally to be there and feel what is actually happening and participate in the signing. And that is what I really feel that should happen in this particular case. When you people come together, you witness what we are doing, and we interact. You see what actually is there, and you advise, and we also show you what we have actually done. And I'm very glad that this happened, especially yesterday when the IAB went round and had very useful interaction with our KIPRE scientists. And I am sure that the interaction you have had will actually mean much more than all the theories that we will discuss today. The conference represents a unique opportunity to engage with some of the brightest minds in the field. Together, we are here to address the critical issues facing our healthcare systems and share innovative solutions that can enhance health outcomes for all. In recent years, we have faced numerous challenges, some public health emergencies to the increasing prevalence of chronic, non-communicable, and forgotten diseases. These experiences have highlighted the urgent need for effective collaboration and application of evidence-based practice in our approaches. Collaboration and partnerships are the heart of what we do. As we share our research experiences and best practices, let us remember that the solutions to our most pressing health challenges often come from the interaction of diverse perspectives. I encourage all of you to engage openly in discussions, challenge each other constructively, and embrace the spirit of partnership. I would like to take a moment to recognize the tireless efforts of health professionals, researchers, policymakers, and advocates to this cause that we have just mentioned. Your dedication to improving health and well-being of communicable and are vital to our collective and success of whatever we actually aim to do. The work you do does not only impact individuals, but shapes entire communities. As we look forward towards the future, let us be guided by a shared vision of health, equity, and accessibility. Our goal should be to create systems that are not only responding to crisis, but are also proactive in promoting health and preventing diseases. Together, we can build a prosperous environment for all. In our discussions, let us direct our energies to converting our laboratory findings to practical solutions to our health challenges. I know that very often we come up with very good findings in the laboratory. We go and put it in the files, and we forget to actually put them into solutions. And discussing today with Professor Jan Howe, we actually mentioned this question of having findings which actually we get in the laboratory, put into practical solutions, and then they help mankind in that respect. And I think this is particularly important in our environment here. If you go into our research laboratories and our files, you will be surprised how many good findings we have, but which have actually been put in files and kept there. So I would very much encourage us, and especially the scientists in the South, to convert our findings into practical solutions so that we meet our challenges. We do produce quite a lot of products which we have just kept in the file. We go to import some very expensive products from the north and forget the practical things that we have here. There are a few things I could mention, but I think this is not the time for which. So with that, I wish you a fruitful and very successful conference. And would you want to invite her? Yes. And with that, I think I end there and invite our DG to call upon our chief guest to address this conference. Thank you very much.
Thank you very much, Chair. Kiple is privileged to have International Advisory Board, which is composed of eminent scientists across the globe. And this week, we have them physically here. So it's just fair for me to invite the chair of the International Advisory Board, Professor Jan Howe, to talk to you and also introduce the other members of the IAB.
Thank you very much, Director. I shall be very, very brief. I think we are very delighted in the IAB that this is the first time we meet in person. we have met online before, but we have not met in person previously. And we are very, very grateful to the senior management of KIPRA, the Director General and the Director of the Board, to have invited us to not just to meet here, but also to attend this wonderful conference. And I would like to just briefly introduce the IAB members present right now. And so if you would please stand up when I mention your name. We have Professor Tony Perry and Professor Jan Panoise, Professor Steve Shapiro, and Professor Yasmina Paramastri, and Professor Jason, that I'm sure you're all familiar with. So on behalf of all of us, thank you very much for the invitation to Kenya and for the opportunity to actually go and look detailed at all the facilities at Kepra and discuss the way ahead and see if we can contribute to that journey. And thank you very much.
Thank you very much, Professor Yan Hao. We have received a lot of support from the Departmental Committee for Health. And in a very special way, on behalf of Kenya Institute of Primate Research, I seriously want to appreciate the support that we have been getting from the Committee for Health. We are also privileged today to have representatives of the Departmental Committee for Health, led by the chair. It's now my duty and honor to kindly request Dr. Nyikal, the chair of the Departmental Committee for Health, to come and talk to us. Karibu, Daktari.
The Principal Secretary in the Ministry of Public Health and Professional Standards, my colleagues, the members of the National Assembly Committee on Health, the Chair of KIPRI Board, Professor Mulimba, and the board members who are here, the Chair of the Advisory, the International Advisory Board, and those who are here, all the distinguished researchers and the academicians that are here, all the delegates that are here, good morning. Let me also take this opportunity to welcome all our international guests to Kenya, and also to appreciate the team that has put this together, and also for the invitation that has been extended to our Committee of Health in the National Assembly. I think this is really appropriate. Basic research is actually key to human development and actually human survival. In particular, when it deals with animal research, and more so non-human primates, because that's as close as we get. We do realize, all of us in the scientific, the academic world, that biomedical processes are to a large extent the same in many organisms and any animals. As I was coming here, I think the speaker was talking at the cell level and the organelle level. I think at that level, most things are the same. And therefore, when we have research on animals or with animals, particularly non-human primates, we really get the basic knowledge that we need for all the processes that are important for our survival as human beings. It's not only in the medical field that this will be applicable. It will be applicable in the veterinary field as well. It will be applicable in the agricultural field as well, so that knowledge will actually take us far. And therefore, to come and share face-to-face. Professor Mulimba just said that when you have online conferences, there's a difference. There's a difference of interactions. I think on online conferences, you don't shake hands and pass cards and pass contacts. So I think this is extremely important for us. More so when we now link this in the medical research, like in this country, we have Kenya Medical Research Institute that I think will link with this extremely well in terms of the information that is available. Because even in the medical research, like the Kenya Medical Research Institute, they will also actually at times be using animals and the animal processes. But I think there's a distinction in this one, because I think this one, I'm not versed in this, is also important for the integrity and the well-being of the animals themselves on which we are doing the research or with which we are doing the research. So we appreciate as a committee that we contribute, and that toward the end I'll actually introduce my members of the committee who are here. But the important thing in this area actually is the no-do gap. Professor Mulimba referred to it as having information that are in books, that are in libraries, that are not being used. That is a major, major issue. Very often, we have a lot of information, a lot of research, but there is no link with actual application. Those of us who have been in areas of policy making, there are a lot of questions that the policymakers are asking, and they would want to have information. But they may not be aware that actually that information is available and sitting somewhere. And the link between the research that is available and what is needed in application is extremely important. What is often referred to as the no-do gap. You may know that vitamin C, when vitamin C was actually discovered and known to treat scurvy, it took about 100 years for people to actually apply it for the seafarers. So the seafarers were dying of scurvy while vitamin C was known. But just saying you can carry a lemon or an orange to the sea and your seafarers will be okay. So that is extremely important. So as we do research, we should also start thinking of the link between what research is and what our needs are. As Professor Mulimba was saying, the North-South divide is actually a knowledge gap. And how can we bridge that gap? Another important area that will probably link us as politicians is research funding. We sit in the budget committee, and not often that people think that it is very important to fund research. Quite often we are very busy with funding our immediate needs, not realizing that some of those immediate needs can actually be sorted out if there is good research. So it is very important that we look at how is research funded. Again, it's important that the research questions must link with the needs that the society has, so that the research question is not determined by the funding availability. That is why you will have that a lot of research may actually link to questions that are asked elsewhere and not locally. So again, that is another link that I can call upon the researchers, that when you make research questions, what is driving the research question? Is it funding? Is it academics? You want to get a PhD? Once you got the PhD, the book sits there. So these are very, very important. Now, what is the role of Parliament? Why are we here? I think one is funding. We sit in Parliament in Kenya. The Parliament has got the power of the purse. we have the budget committee, and even in our committee, as I will introduce you later, we are linked to that. So we can make decisions if you are well informed on what the research is when we are actually applying the funds. Two, it is important that research is guided by legislation, because research itself can actually be wild and may actually interfere, and we may also need to create relationship between different research organizations. So that will be necessary at the point of legislation. So once again, let me thank the organizers for inviting us here. And we came in fairly a good number for politicians who, we are always busy, politicians are always busy. So here we have with us Dr. Pukose, who is a member of the Health Committee of the National Assembly. He was the chair before, so I taken over from him as the chair of the Health Committee, but he's also the vice chair of the Budget Committee. So when a director and PS, you have him there, he's next to you, so that's why he's here. So he just, because whatever you bring to me as the Health Committee in terms of budget, we'll have to go to the Budget Committee, and therefore he will help us. We also have Professor Jaldesa, who is a member of the Health Committee in the National Assembly. He is actually an obstetrician and gynecologist. Dr. Pukose is a surgeon. I'm the smaller of all of them. I'm just a pediatrician. So, and more so a neonatologist, so deal with the small preemies. But as a team, we are actually committed that we should support research, we should support the ministry. I saw Honorable Kibagendi, he was here with us, maybe he has just walked out. With that, let me just take this opportunity to thank you once again, and those of us who have come from outside Kenya. This is the only city in the world where there's a national park. In this hotel at lunchtime, when you sit, you will be looking at the park. They are actually in the park, isn't it? And you can see lions and other animals just sitting at your hotel. So take time, not just to look at the hotel, but to visit our other parks around. With that, thank you very much, and we appreciate the invitation.
Sorry to come back. I was almost punished because I did not introduce my board. First, I would like to point out that actually our board is fairly young. In fact, it is the first board for Kipre. And I was just telling Professor Yan that you seem to know about Kipre more than us. I'm wondering how long have you been coming? And I was surprised how many times he has been here, how many people he has trained. And we are just a new board. I know the people like Professor Shapiro, Professor Jesse have all been to IPR before we actually came in. So you'll forgive me for not introducing my infant board, which is, please, members of the board, just stand. Let me. We are actually supposed to be 9, but there are just a few of us here. On my immediate right is Ms. Salome Owonda, who is actually one of the newest members of the new board. Yeah. Not only is the board new, but she's also the newest. She has been with us for less than a month. Or am I wrong? Yeah, you are most welcome. Thank you. And next to, you can see it, Salome. Next to Salome is Professor Johnson Kangede. He's the chair of our research committee. And in fact, is probably the most traveled of the members of the board. And he's a professor at the JK UAT. Welcome. And next to him is Professor Jumba. Professor Jumba, of course, those are the veteran members of the board, because they came among the very, very first. And at the first we were very few, but now our number is increasing. I think there is another member of the board who has been appointed, who has not arrived yet. So Professor Jumba, Professor Kangede, and Salome are members. And my name is Professor Mlimba. I was probably the first member of this current board. So that's the board, and we are happy. We have had a very good working relationship with our DG and the other members of the staff, and we encourage you to do the same. And we are very happy. We have worked very well with the Ministry of Health, and the PS has almost been a constant member of our functions wherever we have had anything to do. And we are very happy with the Committee of Health, which you have just seen. Most of them are colleagues. Some of them are even my student. So that's how old I am. Thank you.
Thank you very much, Chair. Thank you very much, Dr. Nyikal. I just want to make one plea to the Departmental Committee for Health. There is a journey we started together for the Kiple bill. And I really want to appreciate the fact that the committee agreed that it should be a committee bill. Kindly help us to complete that bill so that Kiple can be well anchored in law. At this juncture, thank you. At this juncture, I want us to put our hearts together and start up and welcome the Principal Secretary for State Department for Public Health Professional Standards, Madam Mary Muthoni Muriuki, to give the key speech and also officially open the 20th Kiple Scientific Conference. Karibu sana, Madam PS.
Please get seated. Thank you. Thank you very much. The Chair and Board of KIPRE, the members of the International Advisory Board, the Chair and members of the National Assembly Departmental Committee, distinguished scientists, researchers, innovators, development partners, academia, private sector leaders, representatives of regional and global institutions, ladies and gentlemen, good morning. Is it still morning? How is Nairobi treating you? You're just next to the park, so don't be in a hurry. Once you finish, the park is just here. There's a lot of things that you can enjoy in Nairobi. So I must confess this, I am reading on behalf of my cabinet secretary, who has proceeded to Senate. We had been summoned for some few questions that we needed to answer. So he requested me to come here and he proceeded to Senate. So it is a great honor, first, to be here for this function, and equally to be the one representing the cabinet secretary for this conference. So on behalf of government and the Ministry of Health, I want to warmly welcome you to Kenya and to this important convening under the fitting theme, Transforming Health Through Innovative Research. This conference, don't look at it as more than just, I look at it as more than just a conference, a scientific gathering. It is convening of purpose. And here we renew our shared commitment to advance the frontiers of knowledge, convert discovery into delivery and ensure that research meaningfully improves the health and the dignity of every person, not just in Kenya, but globally. Across Africa, around the world, so science must move from the bench to the bedside and from publications to people. In our country, we have become very dynamic and we are rolling out our universal health coverage, and it is robust and it is working. Our health landscape, just like I've said, which is very dynamic, we face dual burden of diseases, which we have persistent infectious threats alongside a rising tide of non-communicable diseases. And we confront antimicrobial resistance, health effects of climate change, mental health needs, and lingering lessons of global pandemics. Yet we also stand at a moment of unprecedented opportunity with breakthrough in genomics, digital health, vaccine science, biotechnology, data analytics, and locally driven innovation. So the government's agenda is very clear. Accelerate progress towards universal health coverage, which is anchored in equitable access, quality of care, and financial protection. To strengthen resilient, people-centered health systems from primary health care to specialized services, and prevention and control of communicable and non-communicable diseases with evidence-based, community-centered strategies, safeguarding maternal, newborn, child, and adolescent health, and protect the vulnerable and marginalized populations. So building of robust capacities for health security, including surveillance, laboratories, and emergency preparedness is the way to go. Catalyze local research, innovation, and manufacturing to secure supply chain and sovereignty in health. So as researchers in this room, you must be prioritizing for transformative and for impact. And as we reflect, if I look at it in the Kenyan context and allow to highlight just a few domains where our work is most powerfully into better health outcomes, we have seen having better health outcomes. Talk of preparedness, One Health, strengthening genomic and wastewater surveillance, rapid diagnostics, and early warning systems. We've seen an advance in One Health collaboration across human, animal, and environmental sectors. And that is where KIPRE comes in handy. We invest in pathogen genomics, bioinformatics, and real-time data, which is integrated with integration for faster and smarter responses. When we look at antimicrobial resistance, then the scale of surveillance and stewardship across hospitals, the pharmacies, the veterinary care, and agriculture, the support locally relevant treatment guidelines and affordable diagnostics to reduce inappropriate antibiotic use. Going to non-communicable diseases again, and mental health issues, building predictive models and pragmatic trials to improve hypertension, diabetes, cancer, and mental health care. Therefore, the expansion to screening and early detection, especially at primary health care, integration of digital tools for continuity of care and adherence of support is equally very important. Again, innovative around respectful maternity care, referral system, emergency obstetric and neonatal care and nutrition. We also have local manufacturing. KIPRE has been at the front line of championing local manufacturing. the advancement of vaccine, therapeutics, diagnostics, and the biologistics manufacturing capacity in strong partnership with academia and the industry is a strong conviction that we are going to make it. The support of technology transfer again, compliance, and end-to-end quality systems in the same. There is need for the expansion of biobanking, the cell, the gene therapies, and precision medicine research where it is feasible and ethical. Let us not forget digital health. At any given time, you won't go far. If you want to be innovative, if you want to be impactful, then digital health, AI, and data for decision making. It is only imperative to know that whereas we really need money to support us in all these innovations, then we may need data to convince whoever is giving us that money. We will need data to ensure that we deploy interoperable, secure digital health platforms to inform policy and equally improve on frontline care. It is important that we have digital health equally to harness AI for triage, for imaging, forecasting, and supply chain with guardrails for safety, equity, and privacy. Issues, climate change and health, the extreme weather condition, not just in Kenya, but across the globe. And this has become a problem to quantify and mitigate health impacts of heat, air quality, vector ecology, drought, and floods. And I'm sure the development of climate resilience health facilities will be very important, and climate-informed surveillance again. Therefore, ladies and gentlemen, to ensure that research transforms lives, we must strengthen the ecosystem that powers it. The ethical, inclusive research, uphold the highest standards of ethics, data protection, and community engagement. Ensure that there is representation across board. Our health sector is a devolved function. Therefore, having our counties on board in whatever that we do is something that is very, very critical for us. Across involvement of all genders, age, the vulnerable groups, recognize and respect indigenous and knowledge and local context. Equally, recognition of talent and institutions, investing in postgraduate training. We may do all this, make plans, big plans, but if we do not look at the healthcare workforce that we have, are we improving on them? Are we sending people for training, in-course training, and equally postgraduate training? Because if we invest in postgraduate training, technical fellowships, and continuous professional development, then we will be able to strengthen our institutions. We are able to strengthen our laboratories, our biorepositories, our clinical trials network, our research governance. We are able to have a team that can move forward and ensure that we achieve that which we have planned. And of course, encourage mentorship. Therefore, the next meeting, we should be double this room of people with similar capacities, similar skills, as others coming for mentorship, so that we leave nobody behind. I'll talk about financing, and that is why I was seated next to the vice chair of the budget making committee, because I have to keep planning how they will keep increasing our financing. So, but on this, we need to talk about blended financing, even if we have grants or donations, we must also look at our internal resource mobilization, in-country mobilization. But I'm also requesting that we go for either public, the private sector, let them come on board so that we are able to collaborate further. Philanthropists and multilateral investments. So we must align investments equally with our national priorities and measurable outcomes. When we talk about financing, we must learn the needs of a country. I have just said that is a country. Kenya, we have rolled out the biggest robust program, the universal health coverage, which we look into anybody who wants to come on board, then we must align into the country's needs and interventions. And finally, ladies and gentlemen, we cannot run away from regulation and quality. As we move forward, we must work closely with regulatory bodies, regulatory authorities to accelerate safe, effective innovation, build capacity in pharmacovigilance, device vigilance, product quality assurance, streamline ethical and regulatory pathways, and ensure that we are maintaining this rigor. That is the only way to go. We don't want to go miles and then one regulatory body raises an alarm on a very simple thing that we could have done before we took to the center stage. Therefore, regulation and quality of products and services must be adhered to. I want to assure you that in Kenya, we welcome everyone to invest in the country, to collaborate with our institutions, to move the country forward, because it is only that when we are safe that also our neighbors are safe. And the global community, in collaboration, we become one. The world has become a small village, because if we are not safe, you could not be seated here, probably. Therefore, let us join hands, and in this conference, I want us to come up with resolutions that are workable. And after this conference, it will be time to move the decisions from the table into action, into implementation, into tangible move, or into tangible resolutions that we can see later. So thank you very much for listening to me this morning. I am happy to see all of you, and I'll be more than excited to participate in any other conference that will be held in the near future. Asanteni sana, and now I want to declare this conference officially open.
Thank you very much, DG, for guiding the session. Thank you for all our visitors who have come to grace our occasion. We feel supported now that the conference has been officially opened by Madam PS. We are so grateful, and now we are free to proceed. I echo the words of the PS, that research must transform life. I think that is really very important. And that is why Kipre has serious agenda, like development of antivenom, snake antivenom. You have had development of interventions for AMR using bacteriophage. We work with communities, and we are here to interact with everybody, and of course, increase the workforce for health. At this time, I'm sure Everybody looks like we need a break. And I'm happy to announce that we have a cup of tea and coffee served. We shall break from here, take 20 minutes, have a cup of tea, and then we shall come back. So we shall walk this way, then we shall be guided where we shall take a cup of tea. Thank you very much, and may God bless you. As we shall be taking coffee, we will have a group photo for memories, a group photo for memories, so we shall have a group photo just outside here. Madam Lusline will guide us.
such a rush, my baby, slow down, come find your love. I'm hoping you see me, come serve it up. Love it when you stir it up. That body belong on TV. Oh, this feeling I can't hide. You're feeling up my mind, and I just don't know why.
Okay, thank you very much. I think we need to proceed with our session. We also need to catch up on time. I know time has moved, but we should be able, by the end of the day, we should actually, before lunchtime, we should be able to catch up on time, because I know we have other presentations that are online, and they will only come at the specified time, so it's very, very important to do so. So I just want to announce that those ones who are making presentations, kindly ensure that your presentation is loaded at the back. Yeah, check the program, ensure your presentation is loaded at the back, so that at least we are able to start on time. We will have a timekeeper. A timekeeper is going to sit here and is going to show you have 5 minutes, 2 minutes, so that we are able to manage on time. So at this time, we are going to continue with our keynote speaker, talk number 2. And our keynote speaker, talk number 2, is none other than my friend, Professor Geoffrey Wahungu. I hope ICT, you are loading the presentation, should be ready. So Professor Wahungu, he has been around. Oh, you are somewhere, you went to this corner, and I'm looking for you on the other corner. So Professor Wahungu is going to give a presentation on implications of climate change on ecosystems, biodiversity, and health. Professor Wahungu is the Chief of Conservation and Programs at Olbacheta Conservancy. He holds a PhD in ecology from Griffith University. He began his career at the Kenya Wildlife Service before moving to Moi University, where he served as chair of department and founding dean of the School of Environment and Natural Resources. He then moved to the National Environmental Management Agency, NEMA. I think when you went to NEMA, you became very famous at that time. As Director General, he has taught as a visiting professor at Colorado State University, McGill, Rutgers, Linköping Universities, among others. His long-term field research on the effect of climate change on the habitats of black rhinos at Olbacheta has spanned over 2 decades, is widely published, and has served in several journal editorial boards. He is a member of the Environment Institute of Kenya and the National Primate Task Force. So let's appreciate him as he comes in front. Professor Wahungu, you are welcome.
Good morning, everyone. There was So, ladies and gentlemen, good morning again. It's a pleasure for me to be here, and I'd like to thank Kipre and the committee that invited me to give this keynote. I looked at the topic that I was invited to come and talk about and thought a bit about it, because if you look at our national situation, Kenya doesn't even contribute 0.1% of global emissions that are affecting climate change. And even then, our national determined contributions for 2035 are to reduce that less than 0.1% to by 35%. So I thought I would talk about something that relates to to the work I've done and have interacted with. So I'm waiting for them to load that so that I can. So today I'll talk about two biomes, or rather two completely different ecosystems: Tana River Gallery forests and also acacia savannas at Opegeta, where I work and where I've been working. It's working now? Thank you. I started my career, and this was a sad morning because I hadn't known. I learned about the death of Jim Ells this morning, and he was one of my mentors very early in my career, took me up as a manager and a research officer at Mucherero Research Center when he was the deputy director at KWS. I just felt that he actually never moved out of IPR then to KWS mentally. But then I started my career at Tana River, and now later I'm working at Opegeta. I have worked at Opegeta doing research for about 22 years, looking at the effect of climate change, particularly drought, on interaction by herbivores. So the reason why I'm talking about NDCs and why I shouldn't be dwelling so much on greenhouse gases is because of our national situation, that we are quite low on that. But then again, we can't actually talk about climate change without talking about greenhouse gases. But the most important thing to me are the amplifiers, which actually accelerate the impact, that is land use change, deforestation, browsing, fire regimes, and rainfall. It's either dry or wet in the tropics and in Kenya, and then everything else works around that. It's either hot or cold, and everything else works around that. So there are some socio-economic and environmental vulnerabilities, and today I'm going to talk about environmental health rather than human health, or implication of environmental health to human health. When we talk about high-level impacts, for example, in the Arctic circles, much more than in the tropics, we talk about rising temperatures. Here we have rise in sea levels, which we witness in some of the areas, greater rainfall variability, and of course, what I've been talking about, changes in flow regime. So today, when I'm talking about Tana River, I'll talk about two things that are affected by climate change. One is sea level rise, and the second one, if you remember what I talked about, Mount Kenya, and the fact that Tana River starts at Mount Kenya, glacier melts. And one of the reasons why I'll talk about Tana River and the primates today is because they have depended for many centuries on riverine regimes of deposition and flow. So we have gallery forests that are developed on levees, but then in the past 30 or so years, because of the flow regime changing, the river shifts. And these shifts in rivers, in River Tana, is causing a lot of problems for the primates and their health. Oh, it's working now. So I'll be talking about how primates are affected, and the most important primates for the Tana Delta that you know about that are highly endangered include the Tana River red colobus and the crested mangabe. The crested mangabe is the animal that I used for my master's research way back those days. But most of the other affected in other ecosystems within Kenya include the Brazza monkey, whose distribution you know in Western Kenya, Samburu, and other areas. The Mount Kenya portal that has not been seen since around 1940 or 1938. The Patas monkey, which when I started my research at Opegeta 22 years ago, I observed it for the last time in 2007, and we have not observed it again for reasons which I will briefly dwell on. And then, of course, more sedentary species that don't move and cannot be able to move away from the vagaries of climate change. And most of these include the gallery forest specialists like the columbines. This machine is persecuting me. Okay. Talking about Tana River, and I know a few of us here, or quite a lot of us, Kenyans have worked here, and some of the figures I'll give here may not be consistent with what you know, because every time we go down there, we come up with different figures. But by and large, when we give standard deviations to these, then we come to agree with each other that there are about 75 forest fragments, endangered primates, many river channels, and oxbows. The river is supposed to have changed its course about 5 times since 1990. There is high level of endemism with high diversity of plant species particularly. It's a Ramsar site, very important, and it has high density of birds, particularly Palearctic immigrants that come and use it as a stepping stone on their way south. So it's a very important basin. And with regards to climate change, I want to refer to a study that modeled climate change at the basin and made some predictions based on climate change models and what is predicted, and talked about possibilities of species surviving there, particularly plants and animals, considering that temperatures perhaps rose by 2 degrees above what we have now, or 1.5. And this study by Jenkins et al. Considered presence of refuge. If temperatures rose by 2% or 2 degrees centigrade, would species survive? And they say the model shows that 75% of plants would survive at 1.5% warming. But then the problem is that the areas that were considered as refuges include areas like Tana River Primate Reserve, which is the only gazetted reserve, but then it's also facing a lot of problems, anthropogenic problems, as you understand. So since I think my first attempt at counting these monkeys with Dr. Tom Butinsky was in 1993, and we can say that the population of the Mangabe has declined by 18%. Dr. Kivai was the last one down there, and in consultation. Then colobus, that one is not so much of a problem because they are quite sedentary. That has reduced by 78%. Mangabes are much more adaptive because, as I said before, they can move and disperse and therefore avoid the vagaries of climate change. But the biggest problem are actually the catalyst, or what accompanies climate change, and that is human-induced or anthropogenic interventions that have reduced the forest by 35%. And they are in very, very small patches, which is of a concern. So fragmentation is a problem, and there is fear that genetic diversity, particularly of colobus, would be affected. But this is very interesting. If you look at the study by Bora et al., very interesting that the colobus genetic diversity has not at all been affected. And that's very interesting. There are discussions about what could be causing this, or how they are able to adapt. Climate change, of course, leads to biodiversity loss. Biodiversity loss leads to ecosystem function and services being affected, and that has implications on humans. Let's look at some of the examples that we can give with regard to this. Let's look at flooding. The gallery forests are maintained by seasonal flooding. So naturally, Tana floods every year, and then the flow outside or beyond the levees maintains a good cover of forests. But over the years, sometimes the floods are so high that they actually cut channels, and then old river channels dry up. The forests that were there and where monkeys were are cut off. They get desiccated, and primates are entirely and completely. Yeah, we are back. So, again, if you are familiar with the invasive species issues, Tana is actually struggling with prosopis. And these are actually occupying water channels or colonizing water channels, and therefore old river channels are also at risk. Then we have the isolated populations of birds and other water-living species that are found in the oxbows, old oxbows, and when rivers move away into new channels, these oxbows dry up, and therefore biodiversity is affected. But the most important contribution to this is drought and floods at the same time, or working in conflict with each other. So we have reduced space for primates, and we have also reduced the quality of habitat for primates. And with regard to impact on humans, anyone who has been recently to Garsen, or somewhere between Garsen and Tana River, you'll find a lot of climate refugees. People have been moved by flooding from the rivers up into the dry areas, and they are settled with tents along the tarmac road, receiving donations of food, and it's a very, very bad situation. People who have been having a living and sitting and farming off the levees. So there is increased conflict. There is bushmeat. In fact, the recent emerging one is on bushmeat on magabes themselves, which are endangered species. And what the government is doing now, the county government is trying to do what is called a cluster settlement program, settling people away from the river. They can farm there but be settled outside of this. So we have seen increased conflicts because these clusters are actually being constructed on corridors where elephants and other animals are moving from Savo to access the river and the Tana Primate Reserve. So conflict is increasing. The second indicator other than floods is sea level rise, which is making River Tana at the delta, the mouth, become salty. So what happens when the flow is low, seawater moves inland. And therefore, some of the communities that were settled at the delta, very close to the mouth and were fishing there on freshwater fish, suddenly find themselves without livelihoods. And you can see some of them, especially one of them called Kalota Brook, has had a history of issues related to trying to restore that. And the communities have taken it into their hands to try, and this is completely futile. So this is an issue of livelihoods, and about 5 sub-counties are actually affected. Then I'll move on to the other side of the mountain, Mount Kenya, and from Tana River, move to Ewasongiro River, which is flowing directly opposite the Tana River. And this is a completely different habitat other than the one on the leeward side. This one is a completely dry biome, Alpegeta Conservancy. I've carried out research here since the year 2002, and we actually do monitoring of the effect of drought on competition interactions between elephant, rhinos, and giraffe on one of the keystone species which is a rhino diet at 75%, that is Acacia drepanolobium. The issues involved here are seasonality of the flow. The river sometimes dries up completely. Phenology shifts because Acacia drepanolobium has a problem when it is completely degraded by elephants and other animals, and therefore we have a problem of recruitment because sometimes entire seasons we have no flowering because of competition. And therefore what happens is that we have reduced carrying capacity for all our diversity, particularly where we are trying to conserve the large herbivores like elephant, giraffe, eland, buffalo, and the others. So what we look at is the Acacia drepanolobium. This is a keystone species for black rhino, and we are a black rhino conservation refuge. How elephants actually interact with the habitat to reduce the carrying capacity for Pegeta to hold black rhino. We have about 182 black rhinos. We are the largest sanctuary in East Africa for black rhino. We are larger than any of also the KWS parks in terms of black rhino. So the research we do here is to try and understand what drought does to mediate the interactions. And we use a model which is on tree growth of acacia. Acacia drepanolobium, or the whistling thorn, grows up to about 8 meters high. And therefore, we look at its recruitment. For the last 10 years, we have monitored plots, about 77 plots, from seedlings to about 6 meters, and then see the dynamics of that with regard to recruitment so that we can see the amount of forage that is available. I will not talk this in detail, but one of the deterrents for herbivory for Acacia drepanolobium is the symbiosis between Chromatogaster ants and the acacia. This protects the acacia from herbivory by elephants and other browsers, and therefore works very well to protect the acacia. And that means the acacia is available for rhino. So we monitor these plots. We've monitored them for the last 24 years. The controls are actually within the sanctuaries. We have fenced bombers areas where we do controls where there is no herbivory, and the results are quite interesting. Drought contributes to about 27% of the loss of acacia. Whereas elephants are a big contributor to the damage, others, for example, giraffe, don't have a big effect, but drought plays a significant role. And you can see that if we can keep, for any reason, if the rainfall can be high, then the damage by elephants and other animals to the habitat would be completely low. And we have also seen that in treatment areas where the acacia is not being eaten, it doesn't matter whether there is rainfall or not, but you can see a difference and therefore see that actually climate change is playing a big role in that. And the most interesting result is about diet switching. And this is very interesting about climate change and animal behavior, especially dietary habits. If for any reason we can keep rainfall at our Pegeta and Laikipia at 75 millimeters per annum, then we are able to have the habitat saved because at that level, there is enough grass and elephants switch their diet from eating trees to eating grass. And that's very interesting. I think I will ask for a minute or so because this, oh, now it works. So we also, those of you who know, we have an integrated conservation program where we are keeping livestock. We have about 6,000 head of livestock and about 10,000 head of all the other animals. And we use an integrated system to clean up ticks from the environment. We used to use fire, but this research showed that fire is detrimental to the acacias. So the issues about disease, I'll give one example because of time, and I'll have a disclaimer here. We have a program where we are trying to save the northern white rhino from extinction. And we are using a surrogacy program and using in vitro fertilization to try and save these species. There are only 2 remaining in the world. And the issue about climate change is that in 2023, the consortium, which is called BioRescue, reported that within 1 hour of a surrogate mother being reported to be ill, it was dead from a virus, a bacteria, Clostridium, which is supposed to have been or coming from some skeletons which are buried in the ground, and these were actually exhumed by rain and El Nino, and they affected that rhino and lost a 70-day pregnancy, and this was going to be a game changer because we were going to see the first proof of concept about in vitro fertilization for northern white rhino. We're still attempting this, and we have other activities that are happening. The other indicator I'll talk about is bushfires, because climate change catalyzes bushfires. For us, we have done research on bushfires here, and we have some very interesting scenarios about this. Burning, we've done controlled burns and seen that that has a reverse effect on acacias. Remember the model I gave you? It affects flowering of the acacias. Burnt areas don't flower. Affects mutualisms that I talked about that protects the trees. And finally, even though burn areas increase seedling densities, these burn areas also are grazing hotspots. So we discourage and try as much as possible to guard ourselves against bushfires and wildfires. And you can see the issues of recruitment in terms of burning. So control areas have better flowering and recruitment. If you look at this, a lot of the time, you'll see that this is just the same area. On my right, you have trees that are flowering. On the left, that's an area where there has been a bushfire, and you can see the effect of that. That's much better there. Flowers and no flowers. So I'll skip that burning on. Now, this is my second last slide, and I'll talk about the whole big model and say that drought mediates invasions of big-headed ants that are actually interfering with the mutualisms, and these mutualisms fail, and therefore acacias are not protected. When acacias are not protected, we have loss of rhino habitat, and we suspect, those of you who have studied Patter's monkeys, I said I saw the last one in Opegeta in 2007. We suspect one of the reasons that patterns have locally disappeared from our Opegeta is because of the breakdown of the mutualism. So it's a very interesting observation. How do we mitigate this? One more slide. We create connectivity. If you look at where our Opegeta is at the bottom, we are a fenced reserve, and we are trying to create connectivity so that animals can move out, because drought is going to be a problem to us. So we are trying to create connectivity, and we use that through corridors so that animals can actually move out, and elephants, and also free up the acacia habitat. During drought, elephants and other animals move into our Opegeta Conservancy. When it's wet, they move out and go out into Samburu. So, how then do we try to mitigate drought? Because we are a conservancy and climate change affects us, we try to do an integrated system where we are keeping livestock, and livestock cleans up the environment. We cannot dip wild animals, but cows come into the environment pick up ticks, go to the deep, and leave the ticks in the deep and clean up the environment for wildlife. I'm done. Thank you.
Thank you, Prof. I realized you are very passionate in your work. So, given a whole day, I think you can talk about your work a lot. So, we want to control time, so that we keep time. We shall be going for lunch at 1:30. Lunch will be at 1:30, and by 1:30, all the presentations should end. So we have cut short the time, and so every presenter, when you are coming, you'll be told how much time you have. And at this time, I wish to welcome Dr. Daniel Chai, who is going to be the session chair. And I also wish to mention that when you are here pointing, don't point this way. It will not work. Point this way, because everything is on the other side when you're using this one. Also, we have F&S, Change Biotech outside there, the companies we are partnering with, and they are our sponsors. During break time, kindly visit them. Thank you very much. So, Dr. Chai, save us on time. And there is a bell.
It's afternoon now. I'd like to welcome you to our first session. The theme will be the primate behavior, ethics, medicine. And my name is Dr. Daniel Chai. I'm the Director for Animal Sciences, Welfare and Ethics at KIPRI, also the attending veterinarian. So I'd like to first introduce our keynote speaker, Professor Stephen J. Shapiro, PhD, is Professor of Comparative Medicine in the Department of Comparative Medicine at the University of Texas, MD Anderson Cancer Center. For the past 37 years, he has been studying the behavioral management of the primate colonies at the Killing Center, which includes the chimpanzees, baboons, rhesus monkeys, squirrel monkeys, and all monkeys. He is the founder of the Primate Behavioral Management Conference and is a past president of the American Society of the Primatologists and the former treasurer of the International Primatological Society. He is a recent recipient of a Lifetime Achievement Award from the African Primatological Society. Welcome, Prof.
Okay, how's everybody doing? You ready to listen really fast? Because I'm going to be talking really fast. Okay, we'll go through it. I'm really honored to be here and to get to talk to you all. I did my dissertation at KIPRI when it was called IPR back in 1983 and 1984. How many of you were born in 1983 and 84? Yeah, not all the old dudes in the front, none of the young people in the back. So I'm really honored to be up here and able to do this. So if we could get my slide, okay. I'm going to be talking to you about the behavioral management of captive non-human primates. And again, I come from the University of Texas MD Anderson Cancer Center. Okay. That's a map of the United States, not really part of my, it's part of my talk, but Texas was right in the middle. There's a map of Texas. It's a very large state. And the red star is where I'm located, between Austin and Houston. And we're the large animal farm, kind of, for MD Anderson Cancer Center. MD Anderson Cancer Center is in Houston. They can't have all those animals there. So I'll just tell you quickly about the breeding and research resources that we have. We have about 400 specific pathogen-free baboons. We have about 350 owl monkeys. We have about 1,000 SPF rhesus monkeys. We have only 37 chimpanzees left. You're going to see some of them in a minute. And we have about 500 squirrel monkeys. So that's what we have. That's what our campus looks like. Based on the time, it's not important that I go through that. It's important that you remember this one sentence. If you don't remember anything else about what I say, remember this sentence. I'm a behavioral manager, and what behavioral management does, it serves the animals and the science. We take care of the animals, we improve their welfare, and we improve the quality of the science. So that's what this is all about. Okay, I'm pointing it in the right direction, as you asked me to. Okay, so I'll just tell you quickly, we have a bunch of people who do behavioral stuff, and we have a bunch of veterinarians. The point of my talk kind of is to try and mix or show you how behavioral scientists and veterinarians can work together to improve conditions for the animals and for the research that you're able to do. And this is a video that I'm not going to show right now, but we do acupuncture with our chimpanzees. We have chimpanzees with arthritis. We have 6 of them that are over 50 years of age. Anybody have arthritis? You're all too young for arthritis, but we can use acupuncture to try and treat the animal's arthritis. So we want to promote their physical health. We want them to voluntarily participate in the things that we do. So we don't want to have to anesthetize them. We don't want to have to chase them around. We don't want to have to grab them. We want them to voluntarily participate in what we do. And that includes examinations, anesthetization, some treatments, and if you're doing research, the acquisition of samples. Okay? And we also obviously want to enhance their psychological well-being. That's my job, basically. And one of the ways we do it is we make environments in captivity that functionally simulate natural environments. Okay, that's another sentence you should probably think about. We want our captive environments to functionally simulate natural environments. We also measure the animal's quality of life with regularity. So hopefully this video is going to play and you can listen and I'll talk about it.
Little teeth, have your hand. Hold. Good boy. Your foot. Have your foot. Good boy. Come on, back up. Target. Little teeth, target. Target. Little teeth. Come on up. Target. Good boy.
Listen for the click.
Let's do that again. Foot. Hold. Good boy. Have your other foot. Oh, good boy. Here you go. All right. You open. 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 Good boy.
So we
Very
train him to
good.
open his mouth for us.
Teeth, tail. Have your tail. Have your tail. Good boy. Hold. Good boy. Have your leg. Hold.
So he's voluntarily presenting for an injection.
Hold. Good boy. Very good.
Okay, normally I would spend a minute or two talking about what that was all about. It's pretty cool, isn't it? But I'm not going to do that right now. I'm going to assign you the task of finding me during lunch or during the rest of this meeting and ask me about the details, because I just don't have time really to go into the details now. All right, let's play this one if we can. That's the same one, so forget it. So when you keep primates in captivity, these are our chimps. This is the way we keep them. You have to check the water system every day.
Jane, go check your Lixit.
So we assign the animals the task
Jane,
of
go check
checking,
your Lixit.
testing their Lixits, which is the watering system. So that's Jane. She has to go check her Lixit. And she has to walk by these guys who she kind of likes, but doesn't really like all the time. She doesn't like this guy because he sniffs her butt all the time, and she's really tired of it. Okay, you can smile a little bit, I think. Now, you see, she checks her Lixit. And now we don't have to go in there. We don't have to do it. She does it. And she gets a positive reinforcement, which is a bunch of treats in a paper bag. And I'll explain to you why we use the paper bag when you come find me during lunch, as I'm eating my lunch out of a paper bag, perhaps. So we do this behavioral management. We do the positive reinforcement training to enhance the health and the physical and the psychological and physical health of the animals. We give them annual physical exams. We get some voluntary blood samples. So hopefully the videos are going to play of those which come next. Okay, let's see if this one will play.
Climb.
Okay, these are chimpanzees. We're going to do a bunch of physical exam behaviors with them.
Good boy.
They're pretty big.
Hand. Hand. Good boy. Good boy. Chest.
So we can voluntarily listen to their heart,
Hold.
right? They're participating.
Good job.
And if you can look closely, they're excited about participating in these
Turn
behaviors.
around. Turn around. Good boy. Good job. Zippy.
And again, the open behavior
Open.
that you saw before.
Open. Hold. Good boy.
Chimpanzees have bad teeth, so we have to do a lot
Jelly.
of dental work.
Come on down here.
And getting them to present their rear end for a variety of purposes
Good boy.
is pretty easy. So that's the rectal thermometer.
Hold.
Well, no one's favorite thing, but the animal is willing to do it. All right, let's see what the next one shows us. Will that one play? So now we're going to administer a shot to an animal.
Good boy.
He has to hold on to that bar.
Ready, Fluffy? Stick.
Good boy. Good job, honey.
Okay, so if you have to administer something, get a
You ready,
sample.
Cotton?
That's really all it is.
Good. Hold.
He doesn't like that cotton ball, so we have to desensitize him to the cotton ball.
Good boy.
And if you talk to me, I'll explain what all these terms mean, but I don't have time right now.
Good boy.
Okay, let's advance to the next slide on this,
Okay, once again.
rather than seeing the end of this one, if we can. Now, this is a way to get a blood sample from a chimpanzee.
Hold.
The chimpanzee has to stick his arm into that sleeve. He drinks the juice out of the juice bottle. The sleeve exposes the cephalic vein so we can get the blood sample that we need. We use a vacutainer. And if you can train this in chimps, you can train it in most non-human primates. I'm not going to say any, but certainly most. So again, that's a vacutainer. This is pretty easy, right? This is how long it takes to get a blood sample from the animal. And we've done up to 7 in 24 hours. And I think he's going to change the tube. There you see he's just drinking his juice, no problem. And changes the tube. And on we go. So if we can, let's see if I can, let's advance this because you got the picture. You're not holding the chimp's hand in there because you can't hold the chimp down. The chimp is holding the bar at the end of that sleeve. That's what the behavior is. That's what you train. So let's advance to the next one if we can.
Okay, so we want to manage social housing scenarios like you see. Non-human primates don't share, okay? Do humans share? Sometimes, right? Sometimes humans share. Non-human primates typically don't share. So if we want to feed our group of chimps some good stuff, we have to figure out a way for them to share, so to speak. And the way we do that, it was something called cooperative feeding. And maybe if we could go to the next slide. Okay, so let's play this video. So here we have a bunch of chimps living in what we call a corral. They don't share very well. In fact, one chases and steals everybody else's food. So that one ends up with tons of food, and the ones at the bottom of the dominance hierarchy end up with relatively little food. So the ones that chase and steal end up getting fat. So you might think perhaps I used to chase and steal in my past. If you're really smart, you climb up to the top, because the fat guy won't climb up there to get you when you get your food. So you climb up there, but then you're dependent on the caregiver being able to throw the food just right. So you can hear a little screaming. He even chased a baby. We don't like that. So now what we do is we train him to sit in one place, which is incompatible with chasing and stealing. So which one do you think we train to sit in one place? The one on the front left who's rocking back and forth, saying to himself, I have to chase, I have to steal, I want that food. We train him to stay in one place, and if he does it, we can feed everybody else their junky cabbage and carrots, and he sits in one place. He moved and we called him back. He doesn't care about that carrot. He gets an apple for staying in one place. So it's more worth his while to sit in one place than it is to chase and steal. This way we can feed everybody, and we don't have to use a lot of extra food. We don't end up with a lot of fat guys. Okay? He gets a second apple because he was really good. Let's move on to the next one if we can. And this is just a baboon doing some foraging. We have, like I said, 400 SPF baboons. Baboons eat everything off the ground, so we give them astroturf, artificial turf, and they forage off the astroturf as if it were the ground. So that's once again a functional simulation of natural conditions in captivity. That's what we're about. So let's move on to the next slide. I clicked my thing, we'll see what happens. All right, so as I said, one of the most important things we do is use behavioral management to facilitate veterinary management. We can do acupuncture, we can do physical therapy, we can form compatible groups so that they breed properly. We want some of our animals to breed. We're involved in a number of research projects that involve the animals, measuring the animal's behavior. So we have behavioral research projects that look at behavior. Jan said that was hard. We can do it pretty well, and we can try and look at quality of life. So maybe I tried to advance to the next slide. Maybe if you could go to the next slide for me. Yeah, I'm sorry. I got a few more minutes. Okay. So let's look at this video real quick. This is an animal doing physical therapy. It had its middle finger bitten off, and it had weakness in that index finger. So all we did was take a rubber band, a lacquer band, and train the animal to reach up against the rubber band as a form of physical therapy. So let's stop this one and go on to the next one if we could. Jojo. This one's not going to play. It's just some monkeys that I work with in Mauritius, so we'll skip that. This is the monkey that are on some pain assessment studies. That one's not going to play, so we'll skip that. And I'm clicking, but it's not going anywhere. So we're always trying to get better. That's our goal, to work with the animals as voluntarily as possible so that they want to participate in what we're doing. We get better data when they voluntarily participate. So I'm just going to click through some slides that aren't going to play. And you guys really need a better clicker. We'll skip this one. We'll skip those two. And I'll end by saying we always have to integrate behavioral management with veterinary management and with research management. We can make things easier, the behavioral managers can make things easier for the people who are doing research, for the people who are maintaining the health of the animals. We do a lot. I couldn't show you all the things that we do. We do a lot, but we can always do more. And an important point is that we're never finished. And I'll just come back to the sentence that I started with. Behavioral management serves the animals and the science. And I'll just tell you that we published a book called The Handbook of Primate Behavioral Management. And we're in the process of putting together a second book, a second edition of that same book. I'll also tell you what, up there? There. Okay, well, whatever. I run a conference at my facility called the Primate Behavioral Management Conference. You've sent people in the past from Kenya. Our next one is February 24th through 27th of 2006. We're taking applicants. If you're interested, let me know, and I'll try and get you hooked up. And we also published a special issue of a journal called Veterinary Sciences that's all about the interactions between behavioral management and veterinary management. And at IPS in July, in Madagascar, we also had a symposium that Mercy participated in, and you can find some of those papers that were given there. So I apologize to you for rushing. I apologize to you for not being able to aim my remote control properly so that it would work. Maybe the next person will have better luck with that, but thank you for taking the time to listen to me. I guess I got, there it is.
Thank you, Professor. Let's give him a better clap. Our next speaker in talk number 1 will be Ms. Teresa Nyawira.
Good afternoon. Yes, wait for the presentation to load. My name is Teresa Nyawira. I work at the National Commission for Science, Technology, and Innovation as a scientist. And before I start, just want to acknowledge and recognize that in my career, the KIPRE has played a part. I have great mentors in this great institution, and I thank you very much. So I'll be looking at the evolving frontiers in the regulation of animal research, and I want to dwell really on ethics. My institution is a regulator when it comes to research activities in this country, and as a regulator, we have we are in charge of accrediting research ethics committees in this country, and we have accredited quite a number in different themes, and the KIPRE committee is one of the specialist ethical committees that looks at animal research ethics. My discussion will be to look at really where animal research ethics is, the issues that they are and the direction that is taking. And just before, again, I move on, is to say that in animal research ethics, on ethics generally, a regulator really has to read widely, to understand what is happening in the other areas, in other corners of this globe, because we are really a global village. Research in the US would affect research, the activities that we do here, because you will have trials, that will come and be done in this country. And so it is important that we get to know what is happening in other parts of the continent, the region, and globally. So just to, as an intro, okay, this is a challenge. Uko, okay, okay. Okay, now we have an estimated about 192 million animals that are used in research, and that is from a 2019 publication. And as I was preparing this, I was looking for literature to tell me how are we doing in terms of in our country. And the research in this country that involves animals will revolve around biomedical, behavioral, agricultural, and even non-medical commercial. And if you have ever been to taken medication or undergone a medical procedure, then most likely you are a beneficiary of animal research, because much of the testing, of this medication and even technologies will be done in animals. So animal research is important. And just to mention a bit of that is that we have benefited quite a lot in the in use of animals, and I'm giving just a bit of those examples. The dolly sheep in the Kenyan league, where we'd call her, a celeb in research when it comes to aging. We have non-human primates that have been critical in the development of medication when it comes to hypertension, and the reason is that, this is a condition where the prevalence will also increase with age, and this happens in the non-primates just as in animals. Dogs have come in handy in the development of, dialysis, science behind that. And we have cats that have also been participants in research when it comes to spinal cord injuries and the development of new therapies on neuroscience. So animal research is quite important and, spans in all these areas, whether it's hepatitis, artificial blood, all these research areas have benefited from research in animals. Now, for ethical principles that deal with human-related research, it is easy because we have the 4 cornerstone principles. We have, the respect for persons, the beneficence, the justice, and non-maleficence. But when it comes to animal research ethics, it hasn't, received much attention. And actually, the 3 R's, which are the very common principles that we use when we are reviewing research in animals, the 3 R's standing for replacement, reduction, and refinement, is out of a publication that was done actually in 1959 by those 2 gentlemen, the Russo and Batch. And basically, to run through, because of time, there is the issue of replacement. Replacement would mean that we use alternatives, and with the time, we have quite some replacement alternatives that we can use when it comes to, animal research, that we don't have to use animals, but we can use replacements. We have computer modeling, mathematical programming, in silico methods. We have reduction, where we have reduced numbers, but one problem that we have in reduction is the issue that we can over-reduce, so that the research that comes out of that may not be, the findings may not be translatable because of the numbers. And refinement, where now we minimize animal suffering. Now, apparently, when we use these 3 principles, we have realized that we might even after undergoing the rigors of clearance of such research, then we still have some research activities that are unacceptable going through that. And I want to share with you a few examples that have been published, having gone through the IACUC, the Research Ethics Committees. I just have 3 examples here. And one of them is induced third-degree burns and severe lung injury in conscious sheep, where, conscious sheep undergo burns up to that degree. And we have that publication. We have induced terminal heart attacks in dogs, again, that publication, and learned helplessness induced by inescapable electric shock in rats, and again, that publication, and all animal protocols used in that study were approved by IACUC. I think I want to mention that these are not Kenyan studies. They are, publications in the open journal. But the question is, now that we have the 3 R's principles that, are accepted, is it that not all benefits are worth the cost that they have to, take the animals through? And I note here that even in our own national guidelines as a country, that we also we are talking about adherence to the 3R principles and compliance with regulations. Now, much about the 3Rs and a little about the scientific innovation. We have science is moving fast. The wheels of science are moving pretty fast. And we want to ask ourselves whether these 3R principles, are they sufficient when it comes now to the kind of research activities that we are having going on today globally. I want to give an example of xenotransplantation. Last year, we woke up to news that we have a terminal, a person with a kidney issues that had to undergo some, organ, benefit from organs from a pig. This is, in 2022, again, we had a heart pig getting, being transplanted into a human being. And now what we are talking about is that this is science that will go on. 2 days ago, I read in one of the articles that we have one of the clinical trials using the pig animal organs, to be transplanted to humans. And we can, when you hear a clinical trial happening elsewhere, then you can't be sure as regulators we can wait for those trials right here, because as I said, we are a global village. So again, back to my ethical principles, there are some important ethical questions that we need to resolve. And in the interest of time, what I would say is that scientific developments now call for a better framework. And there are 2 gentlemen who are authorities when it comes to research ethics. One, Beauchamp and Childress, were critical in coming up with the human ethical principles. And now Beauchamp has teamed with another authority in animal research ethics called Gracia, and they have now come up with another framework, and this is what I wanted us also to share. I'm moving to the end of my presentation. And they say that we have seen that the 3 R's, we have the leaky pipe in terms of using the 3R principles. We have the level of pain. Sometimes it's not always, the pain in animals is sometimes not always, looking at it ethically. Sometimes you take the animals through so much pain and you look at the social benefits that are coming out of that. And the question is there a limit? Is there an upper limit of pain that the animals have to go through for the social benefits of animals? And they have looked at all that, and they have come up with suggested these 2 areas that are critical, also integrated some of the principles in the 3R. And I just wanted to share this with you, that 1, the issue of social benefits is important, and looking at is there an alternative method? are there expected net benefits, that there should be significant benefits if we are to use animals to that level, and then also sufficient value to justify the harm. But even as you look at the area of social benefits, it is also important to look at the animal welfare very, very keenly, that there should be no unnecessary harm, at all stages of the experiment. Avoid unnecessary harm. Look at the basic needs of the animals. The fundamental physiological and behavioral needs must be met, and also critical, the upper limit of pain or harm that the animals have to go through vis-a-vis the social benefits. You look at the social benefits and the animal welfare. And so from these reviews, I said as a regulator, you really have to read through. And one of the lessons that I used to conclude is that, one, as we update our regulatory policies, especially when it comes to animal research, it would be important that we recognize some of these developments out there. And then also, as I say, that we still have some research questions when it comes to animal research that perhaps we need to look at. And one of them is, how many animals are we using as a country? Which animals really must be taken through these rigors of regulation and really define what animals that have to go through this. And also the issue that we need to understand what are the public attitudes toward animal research, because it is evolving worldwide, and this will also influence the regulatory frameworks that we are working on. In the morning, the PS mentioned quite some very important emerging areas that as a country we are discussing: the issues of regulatory frameworks for biobanking, the issues of national strategy for precision medicine and genetic research, and all this, and the issue of antimicrobial resistance and use of phages. And now, as we look at this, it is important that we carry along our public, because they are important when it comes to working on our regulatory frameworks. Global animal research will also involve varied regulations with different levels of development that can hinder animal research, and future policy should adapt to emerging ethical and practical challenges in animal research. I hope I did well in time management. Sorry that I had to rush, but thank you very much.
Thank you for keeping time. Our next speaker is Dr. Benson Kibore from Kenya Veterinary Board.
Good afternoon. My name is, been introduced, Benson Kibore from the Kenya Veterinary Board, and I serve as a director in charge of compliance, enforcement, and market surveillance. Thank you for the invitation to this conference. And my topic, on which I'll rush through, is on the regulatory landscape and the role of the Kenya Veterinary Board on animal research. So we are a statutory institution under the Veterinary Surgeons and Veterinary Paraprofessionals Act 2011, and we regulate the veterinary practice, education, and employment. And our other role is to advise government on all matters pertaining to animal health, veterinary practice, and animal welfare. There are a lot of things that I will not talk about because my presenters have elucidated. Oh, sorry. Okay. Now, we are a very old institution. Sorry, I'm trying to rush. I'm pointing at that gentleman. He's telling me to point somewhere. You're not. Is that okay? Sorry. Okay. Oh, yeah. Now I was doing a lot of things. So we are a very old institution of government, founded probably in the year 1953. And ours is actually on the regulation of the profession. And kindly, unless I'm not getting it right. Okay, yeah. Yeah, founded in the year 1953, but we have transformed. Our major role is to register, so in terms of the students, register clinics, hospitals, ambulatories, but also above all is to regulate the veterinary practice in Kenya. So I'll just try to be quick. I'll talk much more about the law, and essentially, sorry, unless I'm not getting it right. Okay, thank you. Yeah, I think that so our functions are that, and we have a role in terms of to regulate the animal welfare. So we register issues of unlicensed institutions that deal with the animal welfare. We advise government on matters of training, research, practice, employment, the use of medicines and animal welfare. We have control over training. And one of our key roles here is to ensure that the institutions that offer animal health and animal welfare actually have the correct competencies to undertake such service. So we do the retention of all the registered persons who must be maintained in the register. I don't want to talk about now the issues that my other presenters talked about on the 3 R's. And I think Teresa, I think, has talked a lot about that. And allow me just to go, I'll not even talk about this one on the ethical committees. Let me go straight to elucidate the interaction between our laws, the regulations, and the international standards, because that is what it is critical. When we make the assessments and check on issues of the animal welfare principles, we check it on the basis of the statutory power that we have, and we look at several issues. Actually, we look at what is it that, and all the designs, all the aims, all the procedures need to respond to the animal welfare. So issues of the methods of handling, the pain management, and all that, they are part of the core of what I'm going to detail in a few. And also issues of euthanasia, because it's a very key area. Yeah, you administer the medications and all that, but at the same time, how you let the animal to rest is what is of very key importance. The ethical considerations and the general health of the animal in the experiment. Now, what we look at from the regulatory point of view, we look at the ethical compliance, and we check at the competency, that is of persons managing the animal welfare. Those are the veterinarians, the animal care staff, the researchers. we check at or we require institutions that have this research, and we check at the clinical responsibilities. Who does what? The post-mortem, the handling, the medical records. Also check at that interaction of the zoonotic and notifiable disease, the pain management and euthanasia. How these animals are sourced, the colonies, the status, the identification, the transportation, because we have statutes that govern all that particular area. And also how you treat that animal within the research facility. So how, and I think my other presenters have done a lot to really ventilate on this. What type of, are you exposing these animals to temperature that are, that will interfere with your results? Too much noise, the animal gets stressed and all that. This interaction between the international standards and our laws, we have also embedded this one in our code of ethics for our professionals, and we check at especially who does the euthanasia, the procedure, medicine used, and what we also say is we check on the professional competence and also the professional incompetence. So we have the two levels that we undertake to ensure that we maintain and ascertain, qualify the animal welfare within our research and laboratories across the country. All these, the Code of Ethics, the Act, is in compliance with the statutory provisions and WOA guidelines, and to ensure that it meets the highest standards of management in handling and pain management. This is in response also, our Code of Ethics, our Act is in response to the principles actually elucidated that as to be undertaken by the committees, or largely on the 3 Rs. I also want to say this, that our Constitution also marks the pinnacle of our laws, and of course it calls for every one of us. One of the things is every person actually has an obligation under our Constitution, and so one tenet is the rule of law. And so one rule of law is that there is also a law that governs the rights of the animal. So as a researcher, you have that individual obligation as a person, as a researcher, to ensure that you observe all the laws that govern the animal welfare. We are in the process of coming up with an animal welfare, animal protection and welfare bill to ensure that we cater for the new inventions actually within the animal welfare space. Just quickly, that's the Constitution, Article 99. It's our role to protect the genetic resources and what we use or what helps us to undertake our research, and also not to endanger the environment. So environmental protection is very key. I want to point out that in as far as we do our research, and our constitutional obligation also calls that we need to have a clean and healthy environment. So if anyone actually, thank you, if anyone feels that those rights of health and clean environment are denied, violated, or infringed, actually they have a constitutional duty, to make an application to demonstrate actually that there is that violation, and they don't have to express or to show any injury for that matter. So what, in terms of the animal welfare, it's the standards, it's as part of the national government function, but in terms of the animal welfare itself, it is individualized to the either respective levels of government or the researchers themselves and the persons who have been given that authority to do that. So this is a regulatory framework that even our penal code, for example, has something on the animal. So if let's say in the animals under research, you are maiming or wounding them instead of the humane treatment, there is also a provision for that. For the wildlife, there is a provision that requires the research permit. The Animal Diseases Act, Cap 364, also has a provision that gives the Director of Veterinary Services that authority over the if you want to actually administer any vaccine or drug or anything, that information needs to be shared with the Director of Veterinary Services. Our Prevention of Cruelty to Animals Act, Cap 360, also is a cornerstone of prevention of cruelty, and this also extends to the animals in research, so that terrifying an animal in research, impounding an animal in a manner to cause unnecessary suffering, starving, or in a grossly dirty environment, those are also offenses that are set in our statutes and within the international, the war guidelines. And you know that as part of our constitutional dictate that any international law ratified by the country becomes part of our laws. I will say this, that that experimentations, and this is very key, and as a matter of compliance for us, is that persons who undertake experimentations on animals need to have the licenses from the board, and there is the restrictions, and of course there is limitations and what we call leeways in areas that are, if they are contributing to new discovery or it's advancing the knowledge, but all in all, it has to comply with the provisions that persons handling the animals must be licensed and trained. Allow me to say that in conclusion, that we have the statutory instruments to check on the welfare of the animals, and that is exactly what the board is undertaking to assure that the research process, the integrity of the animal during the handling, during the processing, during research is given those particular stated rights as indicated in our statutes. So our function is to ensure compliance with the standards in the humane treatment, particularly in the research, the breeding, transportation, and euthanasia and welfare management by animal use and care committees. So, but generally is to say that the reliability and reproducibility of experimental outcomes is positively correlated to the quality of care of animals. I think that has been very much expounded by the speakers that have come here. So I think with that, because Dr. Chai is the one who is looking at me so much, allow me to end there and say thank you very much.
Thank you, Dr. Kibore. I think we better give him a better clap than that. And should you have any questions, we'll give 10 minutes in the last part of this session. Keep your question, then at that time you can shoot your question. So our next speaker will be Professor Yasmina Paramastri.
Good afternoon, everyone. I'm waiting for the, okay, I'm sure. Okay, so my presentation's title this afternoon is Humane Intervention Point for Non-Human Primate in Biomedical Research. So I guess we all agree that the use of animal in research and other scientific purposes is still necessary. So human intervention point is one way we practice refinement. Okay, so this is the outlines of my talk. So we start, what is the human endpoint? So according to the OECD guidelines, the definitions of the human endpoint is the earliest indicator in animal experiment to severe pain, severe distress, and suffering or impending death. So this is in comparisons with the experimental endpoint, which occur when the scientific aims and objectives have been reached. And what is then human intervention point? So human intervention point is criteria that also similar to endpoint, but it is established earlier. So the intervention is not only euthanasia, but it can also include supportive care and analgesia or providing other methods. So the purpose of this is to protect the animal welfare. So human intervention point is applied to both advancements of the research and enhancements of the animal welfare. So we need to find the balance between the advancement of research and animal welfare, and this to increase the likelihood of the animal reaching the end point of the study with less chance of adverse impact on the animal. So the principle, so one of the example of the principle that implemented under the directive of the EU, for example, the death as the endpoint is a procedure that shall be avoided. So I think we agree on that one. I will go really quick for the next one. So what will be important is the establishing and implementing of human endpoint should be a collaborative effort between investigator, ethics committee based on the protocol approval as well as training requirement, and attending veterinarians. So the use of human endpoint, as I described earlier, is a refinement and must be defined in the protocol, and it should be defined before the start of the study. However, based on the guidelines from Canadian Care and Use, is actually is not only recommended, it's not only set prior to starting of the scientific activity, but we need to do that during the scientific activity through the continuous monitoring. In addition to that, after the scientific activity is completed, we should evaluate the effectiveness of the human endpoint that we set previously to be used as information in our future study. One thing that's important is actually finding a balance between animal welfare and study objective. This is very hard, but I think the principle, we don't want to set the intervention point too early, because one can argue that we may euthanize or pull animal from study too early before they actually provide us with the data or study objective. On the other hand, we don't want to set it too late, where the animal will suffer. Pilot study is one way that has been very useful and effective methods to identify and defining a human endpoint, especially for the novel study where some information is not yet available. So this will reduce the negative impact on the laboratory animal. Protocol review considerations is one of the important steps, but it have to be precise, the definitions have to be precise set in the protocol. So we cannot just say sick animal. So it have to be more specific, such as loss, like 15% body weight or 10% body weight. It also have to define the frequency of the animal observations. Training personnel is one of the important aspects in the protocol review considerations. In addition to that, the response, such as what type of actions and interventions that require to be done when the animal actually reached the human endpoint. And of course, monitoring and documentations will be another aspect that need to be considered. So what are the indicator? Assessment and parameter will be very important. So it should be specific parameter. It have to be study specific. and also very important for the species specific. Another important aspect that related to the trained and experienced personnel to do the monitoring. And so this can be reached by the personnel that can recognize normal clinical sign and behavior before they actually can recognize the abnormal clinical sign and behavior or other indicator. So this is one of the publications that indicate like what type of the parameter that can be used to recognize pain and distress. So it could be behavior, physiological, as well as biochemical. Some of you may be more familiar with also the behavior indicator. So like, for example, in non-human primate, reduce of the feeding and drinking or aggressions may be one way that we can see it. And also the expressions or grimace scale may be used commonly. So this is just like one of the resources available that we can access freely in the internet. This is from USDA website. They actually list some of the very helpful resources from Canadian guidelines, NC3R, as well as the UK joint group. This is, some of you may have familiar and use this body condition score, where the 1 to 5 scale are used, that emaciated is score 1, and then obese is score 5. Another parameter, such as grimace scale, you also may be familiar. It has been available for many species, not only in laboratory animal medicines, but also livestock as well as pet. And there's also a publication has been available for the grimace scale in non-human primate. This is actually pretty recent, I think 2021 or 2022, where we can use the grimace scale such as the orbital tightening, and then the posture, like, and also the lip tightening. Some of you may have used this one before. Another publications also, this is used in Japanese monkey, where the symmetrical, asymmetrical facial expression is actually used as the indicator to assess the acute stress in Japanese monkey. One of the very helpful resources is actually available in publications from the Associations of Primate Veterinarians, where the human endpoint guidelines is actually indicates in these publications. For example, the specific percentage of body weight, usually 20%, or body condition score less than 2 out of 5, as well as like non-responsive anorexia for 4 days, and other stuff such as the severe self-injury behavior. So what is the difference for the practical, what we can do between the human endpoint and human intervention point? So again, we try to see, to find the balance between scientific objective as well as the animal welfare. So the human endpoint and human intervention point is very important to be set and agreed between investigator and veterinarian. So this is, for example, instead of using the clinical sign that to decide to euthanize the animal, we may using a certain type of a clinical sign that we actually trigger us to consult with the veterinarians, increase frequency of monitoring, or even temporary removal of the animal from the study, or providing pain relief or supportive care. Then for other example, then if the clinical sign is persistent or repeated, then we can permanently remove the animal from study. So I think we set the earlier parameter other than euthanasia. Another example, like instead of, so we use 20% body weight loss as a human endpoint, but we actually try to monitor the animals. So when they reach 10 to 15% of body weight loss, then we already start with the supportive care or other procedure. Then I think I can use, so Another example of the study-specific, non-human primate has been used a lot in toxicology, neuroscience, and surgery. I will just go very quickly. So this is the OECD guidelines. Some of you may be familiar that used in toxicological study. They did not indicate specifically for non-human primate, but the list of the clinical signs that they actually have is applicable for non-human primate. This is another, just a quick example. Then sometimes fluids and food regulations still used in neuroscience other than the positive reinforcement. So for example, then the monitoring of the hydration is important, or we can also take a blood to monitor the abnormal sodium concentrations. This is another example that surgery and implantations also commonly used in non-human primate. So instead of waiting until the animal is actually have complications from surgery, we may indicate that when the animal have a dehiscence, infections, or bleeding, we're already starting with the interventions such as corrective surgery or other treatment. This is not a new concept, but there is a recent publications that from the CCAC guideline that indicate that it's actually the other point that we need to consider to have reached a lifetime maximal involvement in scientific activity. So this is usually applicable for the animal that longer live and or used in scientific activity for a longer time, including non-human primate. So in other than euthanasia, we can repurposing of the animal. I will go over that later. Removing from the study, and as well as a retirement from research, which is commonly done now with human primate, I think, for example, in the US. For adoptions, non-human primate may not be applicable in most countries based on the regulations. However, the considerations, especially for repurposing animal, we have to look at the health status, potential of future research, as well as the type of procedure that animal have undergone before, and then the invasiveness of the procedure. So in summary, whenever possible, moribund conditions and deaths should be avoided unless it is no alternative and scientifically justified. Development and the implementation human endpoint is a collaborative effort between investigator, ethics committee, and veterinarians. Human endpoint and human intervention criteria must be relevant and reliable. So last but not least, cumulative endpoint should also be considered in longer-lived species to help for a longer period of time. So these are references that I use for these presentations, and then I also would like to point out, like APV website has been very have a lot of guidelines that helpful for a non-human primate procedure. Thank you very much. Thank you for your attention.
Thank you, Professor Yasmin, for keeping time. Our next presentation will be done by Professor Susan Alban.
That was exciting. Hi, everybody. Thank you for having me. I'm excited to be here now for something completely different. I'm talking about wild primates, and because I need my notes, I'm going to open my own presentation and read my notes along the way. I'm going to be talking about how early life circumstances affect longevity in wild primates and what we can learn about physiological mechanisms from that information. So I'm very privileged to be part of this research team. Am I doing okay? Okay, good. It's up here somewhere? Right there, okay. I'm very privileged to be part of this research team. We've been studying the Amboseli baboon population in southern Kenya for many years. This is a wild baboon population. It's not interfered with by humans. For the most part, there are some conflicts with local people. The project was started by Jean and Stuart Altman in 1971, and it's now co-directed by me and my collaborators Beth Archie at the University of Notre Dame and Jenny Tung at the Max Planck Institute for Evolutionary Anthropology. We've been collecting data continuously since 1971. We conduct near daily observations on known individuals in multiple social groups, about 300 individuals extant at any given time, and about 2,200 total in the database. We collect data on social interactions, on environmental conditions, on birth, maturation, death. We also collect biological samples for the purposes of this talk. The fecal samples for steroid hormone analysis will play a major role, but of course we can use the fecal samples for other things. We do also occasionally DART with the help of KIPRI scientists to collect blood and other tissues. Am I doing okay? Yeah, I'm sorry. Okay, yeah, so our team, we have a very strong collaborative team. Our field team on the left here is on the ground 365 days a year, led by Lilian Musembe and Kinua Warutere. We get excellent laboratory and logistics support at the University of Nairobi by Tim Wango and Vivian Oudu. We get great database support in the US, and we have multiple collaborators in Kenya. They play a very active role at the University of Nairobi, KIPRI, KWS, WRTI, and the National Museums of Kenya. It's really impossible to overstate the importance of these very active collaborative teams. It really takes a village to study a wild baboon population, a wild primate population, in as much detail as we do. So I'm just going to tell you a little bit about baboon natural history. Baboon populations are subdivided into stable social groups that are composed of multiple adults of both sexes and juveniles. Females remain in the group of birth throughout their lives, but males disperse from the natal group first at about 7 years of age and then usually several more times during their life. Both sexes engage in multiple mating in a system that we call polygynandry. And birth intervals in our population are about 1 3/4 years. Gestation, as many of you in this audience know, is 6 months with a lactation period of more than a year. Baboons form strong social bonds with group mates that are essential for survival and well-being. This is a grooming shown here in this picture. And individuals can also be linearly ranked within social groups according to their ability to win social conflicts, as shown in this image here of these two males having a social conflict. So to begin the conversation about adversity and longevity, I just want to remind you that adverse early life environments are known to predict lifespan. In humans, individuals with 6 or more adverse childhood experiences, known as ACEs, died a full 15 years earlier than individuals with no ACEs in this canonical, almost 30-year-old study by Felitti and colleagues. It's a remarkable, strong effect. It's been replicated many times, and Felitti and colleagues proposed a set of potential sociocultural phenomena that might contribute to this cascade, starting with social dysfunction, emotional and cognitive dysfunction, and also involving the adoption of health risk behaviors like smoking and heavy drinking on the part of people who had adverse childhood experiences. But since Felitti's work, am I doing okay? Yeah. Since Felitti's work, similar patterns have been discovered in several non-human animals, including non-human primates and including the Amboseli baboon population. So that demands a less sociocultural explanation and a more physiological, generalizable explanation for the phenomenon that we see in which early life adversity shortens the adult lifespan. So we looked at 6 sources of early adversity in our population that I'm going to describe to you briefly. First, did the animal experience drought in the first year of life, like this infant that I'm showing here? Did it have a close-in-age younger sibling, a sibling that was born more than 1 standard deviation shorter than the average in a birth interval? Did it lose its mother in the first 4 years of life? Was its mother of low social status or dominance rank? Was the mother socially isolated? And was it born into a particularly large group? And in our very first analysis of this type, published in 2016, we found this really rather astonishing effect. The y-axis here is the proportion of individuals surviving as a function of age on the x-axis, according to whether or not they had 0 sources of early life adversity. We just counted them up. The blue line is silver spoon babies, if you will, no early adversity. The red line is 3 or more sources of early adversity, and you can see the stratification is very strong. The hazard ratio is almost 2, meaning that with each additional source of early adversity in this population, the risk of death nearly doubles for adult females at every adult age. So why is this happening? Well, a common physiological hypothesis that would be generalizable across species has to do with the hypothalamic pituitary adrenal axis and the secretion of glucocorticoids. In this model, in this hypothesis, early life adversity is associated with an adult phenotype that's unusually reactive, that is especially sensitive to environmental challenges, with the consequence that during adulthood, there's a lot of glucocorticoids secreted into the bloodstream compared to an individual who didn't have that early life programming. The result of these higher circulating glucocorticoids is then sort of thought to produce an eventually caustic, pro-inflammatory, and immunosuppressive mix that shortens the lifespan. Now, some of you think about glucocorticoids all the time, some of you don't, so just as a reminder, I'll just remind you that glucocorticoids are steroid hormones secreted by the hypothalamic pituitary adrenal axis, the HPA axis, and they regulate or support lots of different functions: immune function, cardiovascular function, metabolic function. They are absolutely essential for life. I'm about to tell you some ways in which they're not so good for you, but it's also true that without glucocorticoids, we would just be puddles of jelly on the ground. We would not be doing very well. So glucocorticoids are essential, but like anything, there is a trade-off. So evidence for the first part of this hypothesis is actually fairly robust. Data from humans and non-human animals supports the idea that adult HPA activity is modified by early life adversity. And we have data on that from the Ambacelli baboons. What we do in the Ambacelli baboons is we measure the concentrations of glucocorticoid metabolites in fecal samples, and we call this and this is a really well-established protocol. We call these FGC, fecal glucocorticoid concentrations. And for this particular analysis, we had almost 10,000 fecal samples collected from 192 females over several decades. And what we did was we produced a linear mixed model, a type of statistical model, showing that females with early adversity had elevated FGC concentrations relative to females that did not. Females with 1 or more sources of early adversity had 9% higher glucocorticoid concentrations in adulthood. Those with 2 or more had 21% higher concentrations of FGCs in adulthood. And the largest effects in our data set were for drought, low maternal dominance rank, and large group size. So evidence for the second part of the hypothesis has been quite limited. And we think that that's because this is actually a very difficult hypothesis to test. The best that's been achieved in human studies is 2 or 3 morning salivary samples for cortisol collected over a 5 or 10 year period, and then you follow the cohort until they pass on and you try to look at the effect. And the truth is that it's just simply too coarse to get at what the likely outcome is. Repeated GCs over the life course are difficult. So we were able to bring some key opportunities to the table for this. Again, a large sample of repeated measures of glucocorticoids in fecal samples. In this study, we had 14,000 samples for 242 adult female baboons, as well as detailed data on predictors and outcomes. This is what our data set looks like. The x-axis here is age, and each horizontal line represents one of these 242 females, and there's a hatch mark on each line indicating every time we picked up one of her feces and we extracted glucocorticoids from that. Lines that end with a red dot are females who passed before, who died at that point. Other females are still alive. So we took a joint modeling approach in which we simultaneously estimated 2 submodels. The first is just a linear mixed model predicting what determines glucocorticoid concentrations for these females. And the second one is a survival model, taking the results from the first one and using that fecal glucocorticoid concentration as a predictor of survival. So the 2 models are linked via an association structure. In particular, we took the area under the curve of glucocorticoid exposure for each female and used that as a predictor of survival. So the first submodel, time-varying predictors of fecal glucocorticoids, lots of interesting stuff here, some very unsurprising things like the fact that pregnant females have elevated glucocorticoids. This is a well-known phenomenon. And down at the other end, during the wet season, glucocorticoid concentrations are low because nobody's stressed out. Everybody's got plenty of food. But I won't talk about these interesting predictors. You can ask me later if you want. Instead, what I'm going to do is show you submodel 2, which shows you how glucocorticoids relate to survival over time. Again, I'm sorry, I didn't describe how to read this. As in the previous graph, the x-axis is the parameter value for each of a set of predictors listed on the y-axis. We know that social bond strengths affect survival, so we had to include that in the model. We know that social bond strength of both males and females. We also know that during years of high rainfall, mortality is lower, so we included an annual rainfall anomaly. But the one of interest is the area under the glucocorticoid profile, and you can see that as area under the glucocorticoid profile increases, survival decreases. And to give you a sense of the magnitude of that effect, we've done this visualization, which is of 2 hypothetical females who reach the age of 14. The hypothetical female depicted in blue is one who would have the 10th percentile, the lowest possible glucocorticoid concentrations in our population, and the red is a female who would be in the 90th percentile, and these are model predicted values for the survival probability for those females. These, again, are hypothetical. There's no female who's constantly in the 10th or constantly in the 90th percentile, but hopefully they give you a sense of the potential magnitude of this effect of cumulative glucocorticoid exposure on lifespan. So going back to this glucocorticoid hypothesis, I think we've provided, possibly for the first time, quite robust evidence that higher circulating glucocorticoids are indeed associated with a shorter lifespan. It's an important physiological source of variation that has not been previously documented and that we're very excited about the possibility of looking at in other species, but it's going to be hard. It's very difficult to get those kinds of data. So I'll just end with acknowledgments and thank you for your time and attention.
Thank you, Professor Susan. Let's give a better clap. Our last speaker for this session, talk number 5, will be Dr. Mercy Akinyi from Kipre.
Oh, wait. I need to figure out where this is working from. Okay. Sorry about that. Great. Okay, great. I'll try and do this talk very fast. I realize I'm standing between you and lunch, so I'll not make it a traumatic experience. It's a pleasure to speak after my PhD advisor, Professor Susan Alberts, and she's actually set the stage, thank you, for me to continue from where she's left. So what I'll be talking about is the validation of insertable monitors to measure cardiac responses to stress in baboons. And this work is a collaborative project that was done with multiple partners from different institutions. Professor Beth Archie, right at the top, is from Netadem and was the PI for this particular project. But we also collaborated, of course, with Susan and Dr. Herman Ponsa and Tim Lasky from Medtronic and the University of Minnesota. And as you can also see, there was a huge KIPRE team behind this project. So let's talk about the stress response a bit, though Susan did a good job at that. And what happens is when we get a stressful event, I was hoping the pointer would work, but anyway, from the picture, you'll see that from the brain, you move to the pituitary gland, where the hormones are released that go to the adrenal glands, and cortisol and adrenaline are released to the bloodstream, which then has downstream effects to different organs, including the liver that converts glycogen to glucose. Then there'll be increased blood pressure, fast breathing, accelerated heart rates, and more of slow digestion. And generally, the stress response, as we can see, will focus on the heart. And what is important is the cardiac measures of stress, that is heart rate and heart rate variability, are extremely sensitive and have often been linked to human cardiovascular disease. And these two measures are actually good at measuring or capturing short-term stress responses. And normally when we have exposure to a stressor, there are different paths that this may take. One is the sympathetic system of the autonomic nervous system, which then gives a fight or flight response. And this leads to an increase in the heart rate and a decrease in the heart rate variability. Heart rate variability is defined as variance in time between beats. In contrast, we have the parasympathetic branch of the nervous system, and this is more of a rest and digest activity, leading to a decrease in heart rate and in an increase in heart rate variability. So Susan set the stage by talking about harsh early life experiences and how these have been associated with mortality as well as disease risk, including chronic heart diseases. So these different adversities plus other stressors in the environment can easily influence the cardiac rate and rhythm through the particular branches of the autonomic nervous system that I previously spoke about. So we do have interesting devices, one of which is the Medtronic's implantable loop recorder. And these particular devices are really interesting because they're very small and they're put subcutaneously in the body, and eventually they're able to transmit the data to patients, to particular doctors who then can know how to deal with patients. And these particular devices have been used in animal conservation and welfare. One of them is that they've been put on hunting bears in the US, and they look at the anthropogenic stressors of these animals, such as effects of drones, effect of hunting, crossing roads, and how this affects the heart rhythms in bears. But also in particular zoos, like the Great Ape Heart Project, they do put these monitors to ensure that these animals are in good health and to have interventions for the same. So they've been used before. So what did we want to do with these implants at KIPRE? What we wanted to do is to find out if these implants can actually measure these cardiac measures like the heart rate and heart rate variability, amongst others. But the longer term goals were actually to use these implants at KIPRE and Amboseli to understand what conditions and events are stressful to baboons. As Susan mentioned, a lot of the work that is done in Amboseli is looking at early life adversity and just understanding what's going on. So what did we do at KIPRE? We did deploy monitors to 10 adult females, 5 yellow baboons, and 5 Anubis. And we did use a small little device known as a cardia ECG machine that just helped us know where to put the implant. And we put them at 2 different sites that are shown in blue, as you can see. right at the front and one on the side, just because we thought it would measure the heart rate differently. And the general procedure are actually shown in the pictures at the very top. We have us making an incision afterwards, then using an insertion tool to put the implant, and eventually plunging the implant and covering with a bit 1 to 2 sutures, and of course giving an antibiotic cover. So we also housed these animals socially because we wanted to monitor their behavior, and we also wanted to do other assays. Part of the behaviors that we looked about are agonism, which is really aggression, there was submission, and affiliative behaviors such as grooming. We did something called the human intruder assay, and what happens for this assay is that a human intruder is introduced to the animals, and then the human intruder makes contact with the animals, and then they withdraw. And you as a researcher, you try and find out, was this a stressful experience for the animal? So to go to the results, one of the good things about these implants, because of how small they were, the animals tolerated them well, they healed quickly, the implants did not interfere with the animals' activities, all the sutures were intact, as you can see in the photos. And the other thing is that the implants were extremely accurate, like we were able to get what we expect of an ECG reading. So we did get very strong signals, which showed that the implants were doing a good job at getting our cardiac measures. So some of the interesting things I mentioned about the human intruder assays, and what we did is we had 2 sets. There's the set of staff that normally work towards sedating animals, so they are perceived as high-threat intruders, and mostly these are the gentlemen. But then the ladies are perceived as more low-threat. They're more friendly, pretty much. That's how the primates perceive them. And what we could see on the x-axis is a heart rate, and on the y-axis is the heart rate, and the x-axis is before the intruder shows up, when the intruder is present, and after the intruder. And we can see from the first graph that there are higher heart rates when an intruder is present. That's just generally. But when we look at high versus low threat intruders, we see that the gentlemen posed a higher threat to the animals, and the animals reacted to it. But the also other interesting thing is that we showed that different behavioral observations had different responses. So the red graphs represent agonism, which is like aggression, and you can see the heart rates are higher for those particular instances. But the blue, which is the farthest in the graph, these are more affiliative behaviors, such as grooming, and we can see that for those, the heart rates were lower than those that are associated with aggression, meaning that the devices are actually picking up real-time what's going on the animal. But then it was also interesting that individuals vary in their heart rate responses to social stresses. For example, if you look at the graph, individual with blue, you can tell that 10 minutes before aggression, where the heart rate was, after aggression, the heart rate really went up high, but 10 minutes later, the heart rate still remained high. But then when we can see, we can pick individual that is, I think, brown in color, and for that you can see before the stressor, the heart rate is a bit low. After 2 minutes after, it's very high, but 10 minutes after, the heart rate is able to come down. Then we can see the individuals in green who really, it didn't matter much. The heart rate kind of remained at the same place. So it also just tells you that all of us have different responses to social stressors. So based on the fact that we were able to get this preliminary data at Kipre, then we went to the field in Amboseli to then deploy this in the wild baboon populations. And currently these devices are collecting data. We hope that we'll see something that connects the early adversities that, life adversities that these baboons faced, and seeing if that will connect to the data that we are collecting on heart rate and other cardiac measures. So overall, this study confirmed that the cardiac monitors are a sensitive tool for detecting cardiometabolic issues in baboons, and this also shows that baboons are a valuable model for studying conditions that affect heart health in humans. And I think at Kipre, we do have a department of non-communicable diseases, which then sets up the stage for improved diagnostic tools or development of diagnostic tools, but also development of drugs. This particular tool, the cardiac implants are very expensive, about 6,000 USD. And so if there's room to develop different other devices, that would really be good. And that way we can measure the stress that some of us have, including those who are told to present in 10 minutes instead of 20. Anyway, so that's the general team, and there's a lot of work and a lot of people who contributed to this work, and so I appreciate all of them. And these are the particular groups of other people who are also working both in Amboseli and at Kipre, and this study was funded by the National Institute on Aging. And I think I kept my time. Thank you.
Let's give Dr. Mercy another big clap for keeping time and making sure our lunch doesn't delay. We were to go lunch at 13 hours, 10 minutes, but we are late for almost quite some time now, almost 45 minutes. Thank you. A small announcement. We have veterinarians here, and the Kenya Veterinary Board has a desk right where Dr. Kigora is sitting. You are supposed to go and register there. Also, there is a form, a hotel registration form going around. That's for the hotel. Make sure that you sign, put your name and sign. And we'll need to go for lunch. So for the roundtable discussions, can we do it after we come back from lunch? I think from here, I think you are free, go enjoy your lunch. Thank you for being patient. Thank you for keeping time. The speakers, sorry for the rush. I know we've got a lot of information, but we also had to keep time. Thank you very much.